A very good evening to my lovely children out there. I hope you are doing absolutely great, my dearest champions. So, are you someone who wants to get into the examination hall, look at the question paper and say, oh ma'am, this is so easy, I'm getting full on full. Well, if at all you are someone like that, if at all you have that affirmation, the manifestation, the will to give your best, then this video is going to be perfect for you. Because I am going to put in my efforts, 100% efforts. Just imagine your efforts plus my efforts combined, what will happen, my dear children? You are going to boom. Okay, you are going to boom, and I want that. And I want to see that, and I'm pretty sure all of you are watching this video for the same purpose. For me, none of you are in this level. No. All of you are in this level. All of you are what? Equal for me. And I want you all to be equally good on the day of your examination as well. Don't take this unit test on all lightly. All of this marks comes together, and that's how you get your major mark at the end of the session. And that matters for whatever exam you are going to give next in your life, my dear children. So make sure you're putting in 100% efforts, and I am going to put in my 100% efforts. That's my promise out there. And if at all you agree with me, okay, if at all you're in this battle with me, what you have to do is type down in the comment section right now, yes ma'am, I am going to give my best. Can you type that? While typing that, what are you actually doing? You're telling that to yourself. You're promising that to yourself, and make sure you are keeping that promise. Okay? And the first chapter that we will be beginning is sexual reproduction of flowering plants, my dear children. And over here, obviously, for obvious reasons, I would be going a bit fast. Why? Because there are so many chapters. Okay? The video might be going for 2.5 hours, 3 hours. That should not be a concern for you. Your concern should be that, am I able to cover everything? That's the only concern that you should be having. You should be able to cover everything that I am teaching as of now. You can take a break whenever you need, but make sure you're covering this up. It's going to help you really, really well. I'm telling you, you can trust me 101%. Okay? So, the 1 marker are 5 of them that we are going to discuss, 2 marker 6 of them, 3 marker 4 of them, and 5 marker 5 of them we are going to discuss in this particular session. So, if at all we are ready, give me a thumbs up right now, and here goes the question. Before that, thanks to everyone who commented down under the previous video of Principles of Inheritance, who gave down your valuable inputs, who said that yes ma'am, I accept the challenge, and you watched the complete video, who all commented down their answers in the form of homeworks, my dear children. Obviously, I just want to tell one thing, that I am really, really proud of all of you, and I'll be giving you a comment back really, really soon. I'll be replying to you really, really soon. I saw a lot of you there. I was really, really happy, and I want the same thing here also. Okay, so starting with the first statement that we have, Apomixis is the production of seeds from unfertilized ovules. It seems twisted. Let's make it easy. Apomixis is what? Production of seeds. Is it? Is it production of seeds? Yes ma'am. How are seeds produced, my dear children? So basically, suppose this is the ovary. Inside the ovary you have the ovule. Inside the ovule, what do you have? You have the female gamete. Suppose the male gamete also comes together, it fuses. It will be forming what? The zygote. And now the ovule as a whole, it will be known as what? A seed. So the fertilized ovule is known as what? The seed, my dear children. So Apomixis is a production of seeds. The answer is yes or no? The answer is yes. We are making seed. But do we involve fertilization over here? Usually it's the seed is a result of fertilization. But in Apomixis, do you see fertilization happening? No, my dear children. There is no male gamete which is going to come. Remember that in Apomixis, there is no male gamete that is going to come. Usually what happens? Egg is N, that is haploid. The male gamete is also N, that is haploid. Both of them come together to give 2N diploid zygote. Right? This is what usually happens. But in the case of Apomixis, the egg which is there, it is not N. Instead, it is already 2N in nature. So does it need another N coming from anywhere? No. It itself will transform into what, my dear children? The seed, without fertilization. So Apomixis is the production of seeds from unfertilized ovules, my dear children. Okay, here fertilization is not involved, and seeds are also being produced, which is why it is known as Apomixis. Okay, I hope this is clear. It can be considered as asexual mode of reproduction as well, because in the case of asexual mode of reproduction, do you involve male and female gametes to come together? No. So same thing is happening over here also. Here also, A for absent, males are absent over here. Okay. So fertilization is not going to happen. So this statement is entirely correct. Now statement 2, embryos produced from apomictic seeds are not generally identical to the parent plant. Now the embryo which is going to be produced, my dear children, I told you this egg cell which is there, it is soon going to transform into zygote. The zygote will transform into embryo. Agreed? All of this is happening over here, right? Now don't you think the parent plant which produced the embryo, my dear children, and the embryo, both of them would be similar? Because over here, if you have seen clearly, it's just the female gamete that is participating. There is no male gamete which is participating. That means there is no genetic variation. Only one parent is involved. So their daughter, or what do you call the progeny, my dear children, won't it be the same? It would be the same. It would be identical, no? So here it is written ulta. Embryos produced from apomictic seeds are not generally identical to the parent plant. Wrong. It will be exactly like the parent plant. It will be like a clone, my dear children. I told you apomixis is just like asexual mode of reproduction. And in asexual mode of reproduction, we can say that clones are being produced. Not exactly clones, but yes, comparatively if we see, yeah, it's almost the same. The parent and the progeny. So same thing happens over here. So I can say statement 1 is correct, but statement 2, my dear children, is false. Statement 1 is true, statement 2 is what? False. A become your answer for the first question. So who all got your correct answers? So what happens is that you have to check with me if your answers are correct. Remember, this is a revision. This is not a new teaching series. No. That we have done in Sankalp. This is what a revision. I am here to make you recall everything that you have studied. Are you able to recall that? If at all you are not able to recall that, it's okay. Please pause the video. But if you think my speed is way too much, you can go and control the speed in the settings. Okay? Please do that. In that way only it will be helpful for you. That's a genuine concern. Okay? I'll be going a bit fast because so many questions are there to be covered. That's why. Now, select the correct sequence of events in microsporogenesis. What happens in microsporogenesis? There is microsporogenesis happens where? In the females or in the male plant? Well, micro happens in the male plants. Mega happens in the female plants. Okay, so microsporogenesis is the formation of microspores which later give rise to what? The pollen grains. So first thing that is there would be sporogenous tissue. Okay, and all the cells of this sporogenous tissue, my dear children, would be known as what? Microspore mother cell. The cells of the tissue will be known as microspore mother cell. So they are the mother of microspores. That you understood, no? Microspore mother cell. So they are going to undergo meiosis. Okay, the reductional division, and at the end give rise to what? Microspores out there. They are going to give rise to microspores out there, and this is how the process works. Those microspores will soon transform into what? The pollen grains. So sporogenous tissue, then microspore mother cell, then microspore tetrad, then microspores. Ma'am, you didn't talk about that. So basically, when MMC undergoes meiosis, this is MMC, okay? One microspore mother cell, which is right over here. It divides into two, okay? And again, this is meiosis 1. Meiosis happens in 2 stages. I hope you know that. I hope you know that. If you do not know that, it's totally fine. So meiosis happens in 2 stages. This is the first stage. Again, it will undergo division. This cell divides into 2. This cell also divides into 2. This is meiosis 2. At the end, whatever you get, they are 4 in number, and they are together. They are like, I do not want to go alone. I do not want to go alone. So they are all together. You call this as microspore tetrad. Tetrad means 4 of them. So 4 of them you see over here, they are all together, and then later they dissociate from each other when the they enter dehydrates, my dear children, and further the microspores would be all separate, and they would give rise to what? The pollen grains out there. So your answer would be this: sporogenous tissue, microspore mother cell, microspore tetrad, and then microspores. Over here they have put microspores first. This is wrong. Over here sporogenous tissue, then microspore tetrad. Array, where did the mother cell go? It has to come first. No, then only babies can be born. So this is wrong. Okay, I hope that's clear. So what's your correct statement? Your answer would be A. Now going ahead with the next one, the ploidy of perisperm present in beet seed is. The ploidy of perisperm. What is perisperm, my dear children? Always understand one very important thing. When you talk about the ovary, when you talk about the ovule, you should know the structure of it. Otherwise, what will happen? You won't understand a single word. So this is the ovule which is there. Listen to me carefully, okay? And this is the ovary. Fine. Inside the ovary, you have the ovule. Now over here, the ovary thinks that I should be nourishing the, I should be helping in the nourishment of the ovule. So there is something which is known as placenta over here, and the placenta is connected to the ovule with the help of a structure which is known as funicle. Now the point of attachment which is there, it is known as hilum. It is known as hilum. Ovule says, I am feeling very cold, even though I am inside ovary, I am feeling very cold. I need to have 2 sweaters. So one sweater over here and the second sweater over here. These things are known as integuments. Integuments. So inner integument which is inside and outer integument which is outside. Ma'am, you didn't put the buttons over here. This is a free space. Bacha, this is a free space, and this is what you call as micropyle. This is an opening. And this region where the micropyle will be there, you will call this as micropylar end. Now inside the ovule, what is there? There are certain cells like this. Okay, and you call these cells as the cells of the nucellus. What do you call? The cells of the nucellus. Over here, one cell will be there which will be known as megaspore mother cell. Just like how you had microspore mother cell, here you have megaspore mother cell. This is the only one. Okay, in that you had so many cells, right? In here you had so many cells in the sporogenous tissue, and each of them were microspore mother cell. But here it's not like that. Only one cell will be megaspore mother cell. Only this megaspore mother cell will be undergoing division of what you call meiosis and forming what? Megaspore later on, and this megaspore will later form the embryo sac, and the embryo sac will have the female gamete and everything. So later, if I go forward, my dear children, I would be having an embryo sac over here which is having the female gamete and all the other cells out there. The male gamete will come and fertilize with it, and later this will form the zygote will form the embryo. So by this time, usually the cells of the nucellus disintegrates. They just go off. Okay, they what? Disintegrate, they are used up and things like that. But in some plants, my dear children, the nucellus still persists there itself. They are like, we won't go anywhere, we have to see the embryo grow there. Okay, we have to nourish the embryo, we will be here itself. So this particular thing, when it persists, my dear children, you call this as perisperm. You call this as perisperm. So perisperm is made up of what? The cells of the nucellus itself. And all the plant cells, my dear children, what is their ploidy usually? All the plant cells which are not involved in gamete formation and stuff like that, all of them, rest apart from that, all of them are diploid in nature. That is, it has 2n number of chromosomes, right? So here, if you are asked the ploidy of perisperm, so you are talking about ploidy of the cells of the nucellus. It would be what? 2n, and 2n is known as diploid. Diploid. Haploid means half, diploid means 2. So diploid in nature. Now examples of the plants where you have perisperm, you have 2. B square, remember that. One is beet and the other one is black pepper. Children, I am going to give you homework at the end of each chapter, so make sure you are listening to me. Okay, so you have B square, beet and black pepper where you have perisperm, and the answer would be diploid. I hope that's also clear. Now going ahead, some plants such as viola, oxalis and commelina produce 2 types of flowers. Namely, what is viola, oxalis and commelina? They all are the ones which have 2 types of flowers. So suppose this is a sand region over here, my dear children, or what do you call the lower region. So over here you would have flowers like this, and over here you would be having flowers like this. So as you can clearly see, this is open. This is an open flower, and this would be what? Closed flowers. So in viola, oxalis and commelina, you find open and closed flowers, both of them. The closed flowers are mostly near, what do you call, under the surface of the soil, my dear children. So it is usually found near the rhizomes or the roots. Okay, it can be here as well, it can be here also, but towards the lower region, mostly towards the lower region itself. Now what do you call them and what do you call them? You call this one as clistogamous flowers. Clistogamous flowers, CL for closed, you can remember like that. And this one would be chasmogamous. Would be what? Chasmogamous flowers. So here your answer would be what? Chasmogamous in xenogamous wrong, clistogamous and gytenogamous wrong, gytenogamous and xenogamous wrong. The answer would be chasmogamous and clistogamous, D. And this is very, very important. Listen, understand one very important thing. Again, here the flowers are closed, right? So they are closed like this. So they would be having the female part and the male part. So don't you think here, do you think that here they need pollinators for pollen grains to move from here and there? No, there is no requirement of pollen grains here. The pollen grains, sorry, there is no requirement of pollinators here. The pollen grains from this particular anther, or what you call the stamen, would be falling on the stigma. So you do not need pollinators, okay? And here you can be really sure that seed will be formed. Seed will be definitely formed because it's closed. And here there is no option other than not doing pollination, so they do it. So you have pollination, you have fertilization also happening. So seed is there for sure. No pollinators are also required there. So that's all good, no? So what is bad about this? The only bad thing about this is basically the parent, the male gamete is also from the same parent, the female gamete is also from the same parent. So do you think there will be any genetic variation? No, there is no genetic variation. So that is the only bad thing about cleistogamous flowers. Remember that. Okay, and what do you call this type of pollination? Where the pollination is happening within the same flower itself. Within the same flower. So with the one question I am explaining you so many things. Okay, within the same flower and pollination is happening, you call this as autogamy. Here also, suppose this is a open flower, but from here to here if it's happening, it is known as autogamy. Ma'am, here autogamy is the only thing that can happen. But here, is it the only thing that can happen? No, my dear children. Suppose there is another flower which is open here, another flower which is open here. So from pollen grains from here to here it can land. This is known as when on the same plant but there are different flowers and pollination is happening between them, you call this as gaitonogamy. What do you call? Gaitonogamy. Come on. You call this as gaitonogamy. That means two flowers but of the same plant. Now suppose this plant is there is another plant and pollination is happening between this flower and that flower of the other plant. It is known as xenogamy. Always remember cross, no? A cross is for zero. It is going to cross between two different plants. I hope that works. Okay, so your answer would be what, my dear children? It would be D. And I hope you understood. In cleistogamous flower, you just find autogamy. In chasmogamous flower, you can find autogamy, you can find gaitonogamy, you can find xenogamy also. Cool. I hope that's understood. Now going ahead, the correct sequence of embryonic development in angiosperm. In angiosperm, what is the correct sequence of embryonic development, my dear children? So first of all, zygote is formed, male and the female gamete comes together. Then what happens? There is a pro-embryo being formed. After the zygote, there are embryos being formed. Zygote is what? Single cell stage. Once the single cell divides into two, it's known as embryo. But you cannot call it adult embryo. You will call this as pro-embryo. Now the pro-embryo will have a glow up. It was known as globular embryo. And the globular embryo, when you have glow, my dear children, then there is heart formation, or we call heart-shaped formation of embryo would be there. Heart-shaped embryo would be there. After the heart-shaped embryo, you have heart break also. And then you become what? Mature. And then you become mature. So zygote, okay, then pro-embryo, okay, and then there will be glow. And then there will be heart-shaped embryo, and then there will be heart break also, after which you would be mature embryo. So that's how you can remember it. Zygote, pro-embryo, globular embryo, heart-shaped embryo and mature embryo. Your answer would be C. And it looks like this. So this is a zygote. The single cell divided into two, 1 and 2, as you can see over here. If you cannot see, I'll zoom that in. Okay, 2 cell, and then this is the globular embryo. This is the heart-shaped embryo, ulta heart, but yes. And then you have the mature embryo out there. And that's how things usually work in the case of angiosperm, that is the flowering plants. Works. And now comes the turn for the 2-marker question. Are we ready? I hope we are ready, right? Now complete the tabular column given below with respect to the male gametophyte of angiosperm. Whenever you say gametophyte, what do you mean by that? Gametophyte are not gametes. Gametophyte are the ones that are carrying the gametes. Okay, that would be giving rise to the gametes. They are the one that carries the gametes. So in the case of males, is pollen grain a gamete? No. Pollen grain is actually what? A gametophyte. Male gametophyte. And what does this gametophyte has? It has a plasma membrane. It has the plasma membrane, and then there is a continuous layer like this, which you call as intine. Intine. And it is inside something, that is why it is known as intine, and it is made up of two very important things, cellulose and pectin, my dear children. Cellulose and what? pectin. Now the other thing which is going to come is super important. There is another layer which is like this, which is discontinuous. Why I call it discontinuous? Because you see pores over here. Okay, so this discontinuous layer is very important for you to know. It is known as exine because it's outside. It is known as exine, and it is made up of, okay, it is made up of the most toughest material out there in the whole universe now, which is known as sporopollenin. Please remember it. It is known as sporopollenin. Okay, agreed? Done. Now over here, my dear children, wherever you see pores, what do you call these pores as? You call these pores as germ pores. So this is where the sporopollenin is not there. Okay, this is the exact region where the sporopollenin is absent, my dear children. And why do you need pores over there? I am going to talk about that as well. Remember, sporopollenin is the most toughest or resistant material out there. If you put alkali, if you put acid, still it will be just like that. Okay, now over here within the cell we didn't discuss what is there. I told you that it is a male gametophyte, it has to form the gametes. No. So what are the cells involved over here? So there is a big cell which has irregular nucleus. Which has what? Irregular nucleus. I'll write it also. Irregular nucleus. Now what do you call this cell as? You call this cell as vegetative cell. You call this cell as vegetative cell, and inside this big vegetative cell lies a small cell in the cytoplasm of this cell itself. Okay, and what do you call this cell which is very small and it has spindle shaped nucleus? It has what? Spindle shaped nucleus. What do you call the cell as? You call the cell as generative cell. So inside the pollen grain you have two cells, one is vegetative cell, and inside the nucleus, inside the cytoplasm of which lies another cell which is known as generative cell. So this cell has irregular shaped nucleus and this cell has spindle shaped nucleus out there, and as the name suggests, generative, as in it is going to generate something. What is it going to generate? The male gametes, my dear children. So this cell divides into two, and finally it will be giving rise to the two male gametes. So inside one male gametophyte, that is the pollen grain, my dear children, always remember, what do you have? You have two male gametes. So most of the cells, 60% of the flowering plants, they go in this stage itself. That is, there will be one cell, two cell and three cell. One cell, that is the vegetative cell, and two cell, that is the gamete cells. In that stage it goes, 60% of the angiosperms, 40%, rest 40%, they go in, come back. 60% of them go in two cell stage, that is there will be generative cell and there is generative cell and there is vegetative cell. 40% of them, okay, 40% of them, they will be going in three cell stage. It will already divide over here. Okay. So in three cell stage, you have 40%, and in two cell stage you have 60%. Always remember that. Now they have to go from somewhere, no? Where are they going to go? So this pollen grain which is there, it is going to land on the stigma. From the stigma it has to go to the ovary where the ovule is there. That's where it has to go. So for that you need what? A tube-like structure. So over here, what is the use of this vegetative cell? Not just to provide nourishment, but also to provide a way so that the gametes, whenever they are formed, they can go. So this thing that is being formed is known as pollen tube, my dear children, which is formed with the help of vegetative cell, which is why you call this cell as tube cell as well. So through it, they would be, once they divide, they will be moving down through the germ pores, and that's the use of germ pores as well. So I explained you the whole thing about the pollen grain, and over here you have to tell me this answer. Cells of the male gametophyte, you have two of them. One will be vegetative cell and the other one will be generative cell. Okay, here the shape of the nucleus, vegetative cell, as you can see, it is irregularly shaped nucleus, and over here it will be spindle shaped. That's the answer for this particular question. Works? Okay. Now differentiate between geitonogamy and xenogamy. I told you geitonogamy means same plant, two flowers. Then from here to here when it goes, it is known as geitonogamy. So it is functionally cross pollination because two flowers are involved over there. But genetically, if you talk about that, it's the same plant only, the male gamete from the same plant, the female gamete from the same plant. So it will be genetically like what? Autogamy itself. In autogamy, what did we saw? We saw that there is a flower, the female and the male part is there, from here to here it goes. Male and the female gamete is from the same parent, which is why genetically it will be just same. There is no genetic variation. So functionally cross-pollination, but genetically like autogamy. Now xenogamy, if you talk about that, my dear children, here pollen from this particular plant is going to land on the stigma of this particular plant, different plant altogether. So this is true cross-pollination, that is not true cross-pollination. And over here you can see genetic diversity. There can you see genetic diversity? Not much, because it's the same gametes being involved, same parent, same gametes being involved. But over here you have two different plants being involved. So I hope you can write the differences. Please write in points, please put a box over here, put a line over here, and then explain. Okay, so you can say function, over here you can give 3 points. First is this point, pollen stigma of another flower on the same plant, and here stigma on the different plant itself. Second point could be functionally cross-pollination, over here true cross-pollination. Third point could be it does not bring genetic variation, here it brings genetic diversity. So you get 3 points. Okay. Now going ahead to the next question, clistogamous flowers are invariably autogamous. Justify the statement. What do you mean by that? Invariably means they do not have any option. They definitely will be autogamous. Is that true? Answer is yes. They will be, that is completely true. Why? Because it's not at all open. So they do not have an option rather than to undergo pollination like this itself. Okay, so they remain closed. Clistogamous flowers never open. So pollen cannot be transferred to the other flowers. Self-pollination is ensured. Anthers and the stigma are enclosed together, ensuring what? Autogamy, or which is also known as self-pollination. There is no external agents also needed. Pollination occurs without the external help, making cross-pollination what? Impossible. And over here I have shown you certain pictures. Okay, always remember clistogamous flowers, the closed one you find near the roots, near the rhizomes, or just above the level of the soil. That's all. Okay, so these are the clistogamous flowers that are there. They would never open. Chasmogamous flowers, they are at the aerial position. Remember the name, aerial. Aerial means above the level of soil. Aerial. Okay, that's for chasmogamous. Cool. Okay. So here one very important thing that you need to remember, there is assured seed setting. That means seed would be definitely formed over here. Assured seed setting, that is a term that you need to remember. Now what is Parthenocarpy? Give an example. Parthenocarpy, we studied about Apomixis, no? In Apomixis what was happening? We were producing seed. Okay, the ovule, my dear children, inside this without fertilization only, what do you call, a zygote was being formed, and which is why when zygote is being formed, you call the ovule now as seed. Right. So over here what is going to happen is that the fruit is going to be developed without fertilization. So for that you need to know one thing, that is usually what happens after fertilization, the ovule becomes the seed. Usually, okay, and the ovary which is there, it becomes the fruit. So we spoke about how without fertilization seed can be made, that is known as apomixis, and how without fertilization fruit can be formed, that is known as Parthenocarpy. Ma'am, why does this happen? Because usually there are certain changes in the hormone level, which is why you do not need seed to be formed. You do not want seed to be formed. You do not want fertilization to happen over here. Still, without that only fruit will be formed. So in this type of plants, what is the variety of thing that you can see is that they would be seedless in nature. Why? Because fertilization, has it happened? No. And fertilization, if it's not there, do you think seed will be there? No. Unless and until apomixis is the case, you cannot say. Okay. So here we do not need to talk about apomixis. Here you are just saying that seed is definitely not formed, fertilization has never happened. Okay, no apomixis also, my dear children, but the ovary has turned into a fruit. This is known as Parthenocarpy, and since fertilization has not happened, this would be seedless variety of fruits that you can see over here. Okay, so Parthenocarpy is the development of fruit without fertilization. Result, such fruits are seedless banana, etc., things like that. Induced by, it can be induced through the use of growth regulators by auxins. Can you induce it? Can you make a seedless variety of fruits? Yes. Ma'am, how? By giving the same hormones which were getting disturbed. Okay, because of some hormonal disturbance you got this, no? If you again bring some changes out there, hormonal disturbances out there, don't you think you can induce from your end? The answer is yes. So seedless banana is one very important example, you can remember that. Okay. So I just told you about Apomixis just to have a comparison. Usually children get confused. Apomixis has S in it. Okay, so it's for seed. You remember like that. And Parthenocarpy, my dear children, it's for fruit formation. That's all. Parthenocarpy, it has T in it, so it's for fruit. Fruit also has T in it. Oh my God. Now, going ahead to the next question, define the following terms: staminate flowers and pistillate flowers. What do you mean by staminate flowers? Staminate means, what is stamen? Stamen is the male part of the plant, right? So, staminate flowers are what? Unisexual flowers. That means a flower with just a single sex. With just what? The stamen. These are known as staminate flowers. Okay, now pistillate flowers, unisexual female flowers bearing only the pistil. What is pistil, my dear children? The female part. This is the pistil with the stigma, the style and the ovary. So when you have flowers like this, you call unisexual flowers like this, you call this pistillate flowers. When you have unisexual male flowers like this, you call this staminate flowers. Okay, now in your NCERT there are certain examples given. Okay, when you go ahead with Microsporogenesis, before that, my dear children, when the female part of the plant is taken up, there are certain examples given and you need to remember them. Bisexual flowers, unisexual flowers, all of it. Please remember it. It is going to help you really, really well. Okay. So I think just a second. Okay. Hibiscus, mustard and papaya, watermelon examples are given. Please go read it. Right now I am telling you hibiscus, mustard, H&M and PW. Okay, H&M and PW. These are the examples you need to do. That's your what do you call research spot. Okay, you have to tell me which of them are unisexual, which of them are bisexual. Okay. Go find it. It's going to be really, really helpful. So this is homework number 1 for the day. This is homework number 1 for the day. It's there in your NCERT. Please go and read it. Similarly, there is syncarpus and apocarpus, or what do you call condition. Syncarpus is basically when all the carpels are together. Okay, apocarpus means the carpels are separated, my dear children. Again, there is example given in the picture, under the picture, okay, about Michelia and about Papaver somniferum. You have to tell me what is what. Children, this is all important. Please go through it. Okay. Now, differentiate between pericarp and perisperm. Pericarp and perisperm. What is pericarp, my dear children? So, the ovary, when it becomes the fruit, the layers of the fruit, the wall of the fruit, it is known as what? The pericarp. Now, pericarp can be divided into 3 things. For example, I am talking about a mango. Example, I am talking about a mango. So, my dear children, the outermost thing that we peel off will be known as epicarp. The thing that we eat is known as mesocarp, and the thing that we throw, okay, which has the seed in it, okay, the thing that we throw which has the seed in it, it is known as the endocarp. So, these are the layers of what? The wall layers of the fruit. Endocarp, mesocarp and then epicarp. Epicarp at the outermost thing. Okay, all of them together you call this as pericarp, my dear children. So, the wall of the ovary develops into the wall of the fruit. So, the wall of the ovary which was there, it develops into what? The ovary is transforming into a fruit. So, wall of it will be developing into wall of this as well. So, epicarp is the outer layer, mesocarp is the middle layer and endocarp is the innermost, the hard membranous surrounding the seed. Seed will have its own layers. Seed is what? Ovule. So, ovule will have its own layers and this guy will have it their own. What is perisperm now? Perisperm is the persistent nucleus. I told you, right? Remember, the nucleus says, I do not want to go. I want to see the embryo grow, right? So, same thing. Persistent nucleus is known as perisperm. It is found in black pepper and beet, B square. So, that's also important. Over here in this particular chapter, you have so many questions which are really, really important, okay, and which I'm not going to cover over here, because everything if you are covering over here, what will happen, my dear children? The class will go on, and it won't be any use for you. You will get confused at the end, because 4 chapters all at once, no, okay. So, take it lightly. Okay, I am helping you with the, what do you call, the repeated questions. But that does not mean you don't have to refer to NCERT. You still have to go through your NCERT and what do you call, grasp things up. So, I taught you once, you remember what I said, you open your NCERT, you read it, you remember it better. Please do that. Okay, now draw a LS of grass embryo diagram and label the following parts. So children, I hope you know that there are two types of embryos, monocot embryo and dicot embryo. So what usually happens is that, I think I should be taking this question later on after explaining you some very important thing. Yes, I will be explaining you this question first and then we will go ahead with that. Okay. So picture of a mature angiosperm embryo sac is given below and answer the following questions that follows. Which cells nuclei of the embryo sac produce zygote and primary endosperm nucleus? So when do you get this particular thing? I told you there is one very important cell which is known as what? Which is known as the megaspore mother cell. Where is it? It is inside the ovule. Okay, inside the ovule you have the megaspore mother cell. So what happens to the megaspore mother cell, my dear children? Wait. This is the megaspore mother cell. It divides into two, undergoes meiosis 1 and divides into 2. Again, my dear children, this cell will be dividing into 2, this cell will also divide into 2 at the end of meiosis 2. And what do you get? At the end of meiosis 2, what do you get? You call this as functional megaspore. So you got 4 of them, but 3 of them, my dear children, got degenerated. You got 4 megaspore, but 3 of them degenerated, only 1 was there, and this is known as the functional megaspore. What do you call this process as? This process is known as Megasporogenesis. So we low-key studied Megasporogenesis first. So formation of megaspores from what? From the megaspore mother cell is known as Megasporogenesis. What will happen to this megaspore, my dear children? It will form the female gametophyte. What is the female gametophyte? I told you, gametophyte are not gametes. Gametophyte are the one that carries the gamete or forms the gamete, my dear children. So forming the female gametophyte, which is known as embryo sac. Which is known as embryo sac, and we will come to this question first and then we will go to the first question that we studied. So we will go line by line. So first we took the megaspore mother cell and it underwent division twice. Okay, this is one division only. Meiosis happens in 2 stages, but it is one division only. So one, it divided like this, again it divided. At the end you got 4 cells, 3 of them degenerated, 1 of them was left behind. This is known as the functional megaspore, which later develops into what? The embryo sac. Okay, so these are the steps. Megasporogenesis, formation of megaspore from MMC. MMC location found in the micropylar region of the nucellus. So my dear children, I told you over here, in the case of ovule, you have a region where it is open, where, my dear children, the integuments are not covering up. This region is known as the micropylar region. Here you will be finding the MMC. MMC will be found in the micropylar region. Not of the nucellus, it will be of the ovule. Now, MMC division undergoes meiosis producing 4 megaspores. Only 1 megaspore remains functional, the other 3 degenerates. And this type of development, where only 1 of them is giving rise to embryo sac, it is known as monosporic development. What do you call it? Monosporic. Only a single megaspore is developing into embryo sac. This is known as monosporic development. Single functional megaspore forms the embryo sac. Remember, only they show monosporic development. Now we have got them. Okay, what happened to the cells of the nucellus? There were cells of the nucellus. They can persist, and if they persist, you call this perisperm. If they do not, then it's okay. Now, my dear children, we are going to see the structure of embryo sac. What exactly happens in the embryo sac? This guy which is there, it is now up here in the ovule, the functional megaspore. Let's see what happens to this. Okay, for that we have to go back now. Yeah, so basically I would have to do something. Okay, so basically what happened, so this was the ovule. Inside the ovule you had the nucellus cells and the megaspore mother cell divided. You got 1 functional megaspore, right? This functional megaspore has a nucleus, right? This nucleus divides. Only the nucleus divides. So once it divides, you get 2 nucleus, right? This division is known as first mitosis. This is what? First mitosis. I am talking about what? The functional megaspore. I am talking about the functional megaspore. It underwent first mitosis, you got 2 nucleus. This again undergoes second mitosis, you get second mitosis, you get 4 nucleus. This again undergoes mitosis 1, 2, 3, 4, 5, 6, 7, 8. You get 8 nucleus. You get what? 8 nucleus right over here. Now what happens to this nucleus? And always remember, how many divisions have taken place? 3 mitotic division take place. 3 mitotic division mitosis, not meiosis, okay? 3 mitotic division takes place, and that's how you get 8 nucleus. Now what will happen to the 8 nucleus? The 8 nucleus, my dear children, are surrounded by cell wall now. Okay, so 1, 2, 3 of them will be there over here. They are known as antipodal cells. They are known as antipodal cells. Now 3 of them would be here, 1, 2 and 1 over here. These 2 cells are known as synergids, and 1 cell which is out there, it is known as egg cell. It will be known as egg cell or the female gamete. Now you call this whole structure as what? Female gametophyte, that is the embryo sac. Now what will happen? My dear children, you see all of the nucleuses are there, which are in green color. Here the nucleus are just lying there. So 3 plus 3, 6 nucleus we have covered it with cell wall. But 2 nucleuses which are out over here, they are in one particular cell itself, and you call this cell as the central cell, and you call this nuclei as 2 polar nuclei. What do you call this? 2 polar nuclei. So in total how many cells are there? 1, 2, 3, 4, 5, 6, and this is 7. How many nucleus is there? 1, 2, 3, 4, 5, 6, 7, 8. 8 nucleus and 7 cell condition is there in the embryo sac. I hope that's also clear. Now my dear children, what will happen? I told you in the pollen grains you have 2 male gametes, right? The generative cell divides into 2, you will have 2 male gametes at the end, whatever you do. Now 1 male gamete will be coming and fusing with the egg cell. It comes and fuses with the egg cell. How many, what is the number of chromosomes are there, or what is the ploidy of there? Gametes always will be NN, haploid in nature. So the megaspore mother cell, it underwent meiosis, right? It underwent meiosis. Meiosis is what? Reductional division. Okay, you are reducing the number of chromosomes. So over here, when it underwent meiosis, it started with 2n number of chromosome, but it ended with n number of chromosome. So this particular cell which is there, this embryo sac which is there, all of the cells n will be having haploid number of chromosomes. But central cell, here both the nucleus, 2 nucleus are there, each of them are having nn. So this central cell will be the only one which will be 2n in nature. Rest all of them will be haploid in nature. So this haploid cell, that is egg cell, another male gamete also which is haploid, comes together to form diploid zygote. This is the first fusion that happens, and this fusion that happens, it is known as syngamy. It is known as syngamy. Now similarly over here, my dear children, the central cell which is there and the male gamete, okay, the central cell which is having 2 nucleus, my dear children, and the male gamete which is having 1 nucleus, n plus 2n gives what? N plus 2n gives 3n. This would be triploid. This would be triploid in nature, and what do you call this? You call that as endosperm. You call this structure as endosperm. So central cell and the male gamete comes together to form the endosperm, and it will be triploid in nature. Cool. And what's the use of this, and what do you call this fusion as? You call this fusion as triple fusion. Why? Because 1, 2 and 3 nucleus are involved, which is why you call this as triple fusion. And what is the use of endosperm being formed? Endosperm is the one that will be providing nourishment to the growing zygote. To the growing zygote, so that it can become what? Embryo in the future. Okay, it will be helping in the formation of embryo in the future. I hope that is clear to you. And this fusion, since it is happening twice, my dear children, you call this as double fertilization. What do you call it? Double fertilization. So over here a pollen tube would be coming in, and from over here synergids, they have a structure which is known as filiform apparatus. It will guide it. It will guide it towards the egg cell. And you know what? The best friend, synergid cell, it will die off. It will get degenerated when it allows entry through it. The male gamete has to meet the egg cell. The egg cell is over here. So one has to sacrifice itself, and that will be one synergid. Who shows the way? It is the filiform apparatus that shows the way. So with this, I think so many questions would be clear to you. One is, picture of the mature angiosperm embryo sac is given below. Answer the questions that follows. Which cells or nuclei of the embryo sac produce zygote and the primary endosperm nucleus? What is the answer for this question? What is the answer for question? The egg cell fuses with one sperm nucleus to form the zygote, and the two polar nuclei fuse with the another sperm nucleus of the male gamete. Please write the male gamete. That would be nice. Okay, male gamete to form the primary endosperm nucleus. Primary endosperm nucleus is being formed, or the primary endosperm. You can say the primary endosperm itself is being formed. Okay, since you're talking about nucleus, that's why you name primary endosperm nucleus. If you talk about the cell, the central cell and the male gamete cell, it fuses to form the primary endosperm. That's all. Primary endosperm cell, PEC. Now, what is the ploidy of the following? Antipodal cells. It is what? It's a normal cell out there. It will be haploid in nature. It would be haploid. What is the ploidy of primary endosperm nucleus? Primary endosperm nucleus, my dear children. What would be the ploidy of that? 3 nucleuses are coming together. So it would be triploid in nature. 3N. It would be triploid in nature. Why does endosperm development precede embryo development? Why do you think endosperm develops first before the embryo? Because the endosperm wants to give nourishment. It is a nutritive tissue. It will provide essential nutrients to the developing embryo. That is why it has to be formed first before the embryo is formed. I hope that is also clear. Now, one more question was there. What is double fertilization? Describe the fertilized embryo sac with neatly labeled diagram. You can describe like this, okay? And what is double fertilization? I hope now you know that. One fusion happens over here, second fusion happens over here. These two things are known as double fertilization. Syngamy plus triple fusion. That's all. Fine. Now we will come to this. Where is it? Yeah. Now, my dear children, listen. So, whenever you say an embryo is formed, along with that, one very important thing is there, which is known as endosperm. So, endosperm is that something that helps the zygote to form the embryo, right? But when the embryo is formed, it says that I don't want endosperm now anymore. Instead, I will be having my own ways to nourish myself. So, the embryo which is there, it forms cotyledon. With the help of what? With the help of the endosperm. It takes the nutrition from the endosperm and forms the cotyledon. In some of them, the embryo forms 2 cotyledons. In this case, you call them dicot. In some of them, it forms 1 cotyledon. In this case, you call them monocot. Okay. When they form 1 cotyledon, they will still have some endosperm left behind. So, over here, you will have 1 cotyledon plus little amount of endosperm also in the case of what? Monocots, my dear children. Now, you are asked to make the longitudinal section of the grass embryo. So, over here, why did I tell about all this thing? Because I wanted you to know that grass comes under what? Monocots. They would be having 1 cotyledon and they would be having little amount of endosperm still persisting out there. And the easiest way to remember this particular diagram goes like this. So, over here, this is the embryo. Embryo always has axis, okay? This is something that is going to give rise to, this is something that forms the plumule. Plumule is which gives rise to the shoot later on, okay, the stem part of it. And there will be radicle which gives rise to root, okay. Now, this is covered by something which is known as root cap, okay. It is covered by root cap. Now, since it does not have 2 cotyledons, I will just close this up. There will be just 1 cotyledon which will be like this, and this cotyledon has a name as well. Since there is only one, they have kept a name that is known as scutellum. The rest of the space will be taken up by the endosperm. So, this is what the persisting endosperm which is there. Now, this is a monocot embryo. The plumule is actually surrounded by something which is known as coleoptile. So, near the plumule you have the coleoptile, okay. Over here beneath the radicle you will have something else which is known as coleorhiza. This is known as coleorhiza, and that's how your whole monocot embryo is being formed. Now in your NCERT, they have given a twisted picture. Okay, they have given a twisted picture which makes you confused, but I am here to help you out. So my dear children, I told you there is an embryonal axis, and this embryonal axis will be having a shoot apex which is known as the plumule. It will be covered by something that is known as the coleoptile. Over here you will have the radicle, the root cap and the coleorhiza, right? And now there will be one thing, that is the cotyledon out there, which is known as the scutellum, right? Where is the other endosperm, ma'am? That is not being mentioned in this particular picture. Now what is this epiblast, ma'am? Actually it tried to form the two cotyledons, but it was not able to. So this one is the rudimentary cotyledon out there. It was not fully formed. It is known as rudimentary cotyledon. Now if you look at the picture, I think it will be easier for you to remember it. You have to practice it. You please draw and practice it, but you have to do that. It's for you guys. Okay, do that. Now going with the next question, draw a label diagram of transverse section of young anther. Okay, when you form the anther, my dear children, what is there? Young section of anther, always remember it looks something like this. Okay, at each corner you have what? You have the microsporangium. This is known as the microsporangium. And now you have to make the walls of it. So my dear children, the outermost wall that you see is known as epidermis. So I am going to make and show you one so that you understand it better. So this one is what? Epidermis. Now after epidermis comes what? Endodermis. Okay. Now I am going to make one very important thing. This is the sporogenous tissue. What is it? Sporogenous tissue. And the sporogenous tissue is where you have the microspore mother cell. Remember, they have all microspore mother cells. Now, providing nourishment to this sporogenous tissue, you have tapetum. You have a layer which is known as tapetum. And you see a gap over here. This gap is filled by middle layer. Filled by middle layer. So you have to, what do you call, label all of it. So epidermis, then comes endothecium, then comes middle layer, then comes tapetal cells, then comes sporogenous tissue. So EEMTS. Okay, and the sporogenous tissue, my dear children, they contain the cells that forms what? The pollen grains later on. So that would be the transverse section of young anther. And the tapetal cells, it will be usually binucleate. It has two nucleuses. Why? Because probably karyokinesis took place. Okay, this nucleus divided, but cytokinesis didn't take place. That is, cell did not divide. So a cell can have multiple nucleus as well. Okay, and that is for nourishment. It has enough amount of cytoplasm also, this tapetal cell. It is for nourishment of what? This sporogenous tissue. Always remember that the cells of the sporogenous tissue are nourished by the tapetal cells. The outer cells that you see over here, they are responsible for the dehiscence of anther, for the breakdown of anther. Cool? Yes. Right, any four characteristics of flower pollinated by wind. Water, false fruit, give an example. So you should be knowing all the features of wind pollinated, water pollinated and insect pollinated flowers. I hope you do not, if at all you do not know that, please go and read about that. It's very, 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 very important. Okay. So wind pollinated requires the pollen grains are light in weight. The pollen grains should be light. This is one feature. Okay, now they are should be carried by wind. If it is very heavy, can it be carried by wind? No. It should be non-sticky, my dear children. If it sticks, it will go and stick somewhere. No, it should be non-sticky and powdery so that they can be transported easily by the wind currents. So for pollen grains, we found 2 points. Now, they often possess well-exposed stamens so that the pollens are easily dispersed into the wind. So the stamens are the ones that produce the pollen grains. So they have to be well exposed. Otherwise, how will the wind carry them if they are lower down, right? So that's for the stamen. The flower must be feathery, must have feathery stigma to trap airborne pollen grains. Yes? The stigma which is there, it should be feathery. Why? Otherwise, this flying pollen grains which are there should be trapped by something, no? So that's why it should be Feathery. They often possess well exposed, the flower must have feathery stigma. This is one very important point. Now, wind pollinated flowers often have a single ovule in each ovary and numerous flowers packed into inflorescence. Wind pollinated flowers, my dear children, would have what? A single ovule. Why? Why do you think so? They usually have a single ovule because wind pollination is kind of rare. It rarely happens. So you do not want to make more ovule, but if no pollination will happen or no fertilization will happen, all of it will go waste. No? That's why it usually has single ovule, and each ovary and numerous flowers are packed into inflorescence. When you form an inflorescence, there will be numerous flowers in it. That's all. So about pollen, 2 points. One is that it has to be light in weight and it has to be non-sticky and what? Powdery in nature, so that it can move around very easily. About the stamen, it should be well exposed. About the stigma, it should be feathery to capture all these things. Okay, and usually wind pollinated flowers, they would have single ovule in an ovary, and usually if they have inflorescence, there will be so many flowers in it. Example of the plants that show wind pollination. You have grass. And apart from grass, what do you have? You have grass. Apart from grass, corn. Okay, grass and corn, very important example. For each water pollinator and also wind pollinator also, you should know the examples. Vallisneria is example of water pollinated flowers. Water lily and water hyacinth are not pollinated by water. Instead, they are pollinated by wind and insects. Remember that. Okay, so Vallisneria, what is the name? Zostera. Okay, Vallisneria surface ladder pollinated. It is on the surface level and it is pollinated by water. And Zostera, it is in the submerged condition. It is pollinated by water. So you should know all the examples. Otherwise, it is going to bring out a lot of take away a lot of marks out there. So don't do that. Now, false fruits. False fruits develop from parts other than the ovary, like the thalamus or pedicel. So usually fruits are originating from the ovary. The ovary is the one that makes it, right? Now suppose the ovary rests on the thalamus, and suppose the thalamus is also involved in the formation of the fruit. Thalamus is also involved in the formation of the fruit. In this case, my dear children, you will call this as false fruit. Okay, so apple is an example of that. Apple forms from the thalamus. Cashew, it forms from the pedicel. Pedicel is this thing. Okay, and there is strawberry. So apple, cashew and strawberry are very important. They are what? False fruits. I hope that's also clear. And now I realize the time is way too much. It's 56 minutes now. I have to speed up. Otherwise, how will we complete 4 chapters, right? So describe the structure of dicot embryo with the help of labeled diagram. So dicot embryo, my dear children. So again, embryonal axis will be there. This is going to be what? Plumule. This is going to be what? Radicle. Radicle will still have what? Root cap. Do they have endosperm still persisting? No. They form 2 cotyledons out of it. So this is the first cotyledon. This is the second cotyledon. Okay. Cool. Now if we enlarge it like this, my dear children, so it will look something like this. This is the embryonal axis. And you have the plumule over here. From here, this is one cotyledon. And this is second cotyledon. Always remember, from the point of attachment of cotyledon above it, you will have the plumule. And this area is known as hypocotyl region. What do you call this region as? Hypocotyl region. Is it hypocotyl or epicotyl? Epicotyl region, not hypocotyl. It is known as epicotyl region. The above one is epicotyl. Hypo means lower. Hypo means less. So this is epicotyl region and this will be hypocotyl region. Okay, works. So there is no coleorhiza, no coleoptile. There is no endosperm also, which is still persisting there. Example of dicots. Example of dicots, the ones that you can break into 2. For example, groundnut. For example, what else? Groundnut, pea. Okay, pea you can break into 2. So all these things are known as dicots. Okay, and all of them are known as non-albuminous seeds. Whenever endosperm is not there, you call this as non-albuminous seeds. So pea, groundnut, all of it which can be broken into 2 are known as non-albuminous. Now wheat, rice, all of it, wheat, rice, coconut, castor, all of them are monocots. They cannot be broken down into 2. You do not say that coconut if I throw I can break down to 2. No, I am not talking like that. Okay, I am not talking like that. I am talking about the embryo which is there. It cannot be broken down into 2. So basically all of them are monocots. What all? Wheat, rice, barley, or suppose barley and castor, coconut, all of them are monocots and all of them are known as albuminous because they still have endosperm left behind. Please remember that. Okay, if at all you cannot remember, write it somewhere. So dicot, monocot. I am actually investing too much time here so that you don't miss even a single thing from your NCERT. Okay. Monocot example is castor, coconut, C square. And apart from that, all the things that we eat, rice, wheat, okay, all of this thing. Now dicot example, as far as I can remember as of now, pea, groundnut, all of it. Okay, chalo. So if you are asked to form grass embryo, what will you form? What will you make? This diagram or the monocot diagram? Grass is monocot. I'll write that also. Grass is monocot. Okay. Now, explain any 3 outbreeding devices in the flowering plants. What do you mean by outbreeding devices, my dear children? The ones that does not allow self-pollination to happen. Because if self-pollination will happen, my dear children, the plant will be like, oh, I am tired. I am getting the same genes, same toxic genes. So suppose this plant is not really good, not really having good traits. So it is again getting pollinated by its own pollen grains, my dear children. At the end, it would be infertile later on. You get it? So it prevents inbreeding depression. Outbreeding devices are the ones that will be helping in preventing inbreeding depression due to continuous self-pollination. Now, what are the mechanisms for promoting outbreeding? First one, time difference. Pollen release and stigma receptivity are not synchronized. Okay, the pollen could be released, but the stigma could not be receptive. Okay, so if the time difference is there, then it's totally fine. In that case also, you can avoid fertilization to happen. Now, spatial separation. Suppose the stigma is somewhere over here. The pollen is somewhere over here. It takes a lot to travel down over here. You get it? So, spatial separation, that is one method of doing this. Now, self-incompatibility. That means basically, suppose the pollen grain is like here. It comes and lands also, but still it won't be able to make the pollen tube. Why? Because the chemical coordination, which is very much required over here, is not happening. There's some inhibition happening. So this is a genetic mechanism. You are not doing anything. Okay, it's a genetic mechanism to prevent what? The pollen from fertilizing the ovules. So first is time difference. Time difference of what? Pollen release and stigma receptivity. If the stigma is not receptive, okay, what will happen? The pollen will just come and lie and disintegrate, my dear children. Nothing would happen else. Nothing else would be happening. So that is one thing. The spatial differentiation or separation is the second thing. Self-incompatibility is a genetic mechanism. That is the third thing. Now, formation of unisexual flowers. Okay. Now, unisexual flowers can be of 2 types. One is basically, suppose I have a plant. I have second plant. Over here you have only the female flowers. Here I have only the male flowers. Okay, this is plant 1. This is plant 2. What do you call this condition as? This is known as dioecious condition. This is known as dioecious condition, my dear children, where there will be separate plant for female, there will be separate plant for what? Males. Males and females. They are separate. In this condition, obviously, autogamy cannot happen, self-pollination cannot happen because the pollen grain producing thing is not here. Similarly, over here, stigma is also not there. So, here definitely I can prevent self-pollination for sure in the case of dioecious plant. Example, ma'am, example is papaya. Please remember it. Papaya, DP, okay? Your WhatsApp DP, you can remember like that. Now, that is where pollination cannot happen. Now, there is one more condition where what is happening is that there is a plant, you have flowers. Now, this flower can be female flower, this flower can be male flower. Here, to some extent, okay, to some extent, you can prevent what? Self-pollination. No, self-pollination can be, autogamy can be prevented. Autogamy, if you say, that can be prevented because both of them are not together. But gytenogamy, can that be prevented? No. Gytenogamy will still happen, my dear children. So that is the condition of monoecious plant. Monoecious means both the sexes are in a plant. Both the sexes, monoecious in one. Okay, they would be, both the sexes would be in one. Now condition can be like this in monoecious plant, or it can be like this also when it has both male and the female. So when this is the condition, I say bisexual monoecious plant, bisexual flowers in monoecious plant, bisexual as in both the sexes are there in a single flower, but this is what, a monoecious plant, or this is a condition unisexual flowers in monoecious plant. When you talk about unisexual flowers in monoecious plant, autogamy is prevented but not gytonogamy. Example, castor and maize. Male and female flowers on the same plant prevents autogamy but not gytonogamy. Okay, but over here in dioecious, what is happening? They are unisexual, completely unisexual. Okay, male and female flowers on different plants, they will prevent both autogamy as well as gytonogamy. So this is preferred. Here also to some extent you can stop, but here to full extent you would be able to stop. So these are the things that you need to remember in the case of outbreeding devices. Okay, now going ahead, picture of, I explained you already this particular thing. Explain the structure of megasporangia. This also I already explained. You would be having ovule. Ovule, this is the placenta. Placenta is connected to the ovary with the help of, ovule with the, this is the ovary. This, wait. This is the ovary. This is the placenta. Placenta is connected to the ovule with the help of funicle. This part which is there, this is known as hilum. Now here you have two integuments. Please remember, here it would be open. This area would be open. This is known as micropylar region, and this region at the opposite of the micropylar region is known as chalazal region. And remember one very important thing, there are some situations where what will happen, the placenta and the funicle are attached over here. The placenta and the funicle are attached over here like this, inside the ovary. This condition is known as anatropous ovule. What do you call this? Anatropous ovule, where the ovule is what? Ulta. This is known as anatropous ovule. And in this condition, if you are asked where is chalaza, if you are asked where is chalazal end, what will you answer? Chalazal end will be still here itself. Even though the placenta and funicle have shifted, chalazal end will be still here. Why? Because the wherever the micropyle is there opposite it, you will have chalazal end. And always remember one thing, where is the MMC located? Here in the micropylar end. Where is the egg cell located? Here in the micropylar end. Where does the pollen tube come? In the micropylar end. Where is the antipodal cells? Antipodal cells are on the opposite side, that is chalazal end. Okay, that's all. Fine. Now, okay, picture related to pollination is given. Will this pollination confirm fertilization? What is this picture? There is a bee, honeybee, and a flower. With this, will this pollination confirm fertilization? The answer is no. None of the pollination can confirm fertilization. Pollination is what? Transfer of pollen grains. Fertilization is what? Fusion of male and female gametes. That may happen, that may not happen. So no, pollination does not always confirm fertilization. Pollination is transfer of pollen grains from anther to the stigma, whereas fertilization only occurs if the pollen grain germinates, forms the pollen tube and reaches the ovule. Now next question, what are the floral rewards provided by the plants to the insects to revisit? It can be nectar, it can be pollen grain itself, because pollen grain is highly nutritious, and shelter also. For example, safe places for laying eggs. Example, yucca and the moth. I told about the story of yucca and the moth, right? So that is an example. So nectar, pollen grains, and also shelter for placing their eggs. Now mention the characteristics of flower in this pollination other than the floral rewards. So flower would be large, they would be colorful, they would be fragrant, they will be rich in nectar. Small flowers, if at all they are there, they will come together to form large inflorescence. Animal extraction to flowers by color and fragrance. Now flower which is pollinated by flies and beetles, my dear children, instead of being fragrant, they would be smelling bad, so that it can attract the flies. Make sure you're writing all of it, it's very, very important. Now, okay, finally, okay, so now PU2, PYQ of 2024, my dear children, which came from this chapter, and I thought this question was kind of confusing, which is why I put it down over here. The question goes like this: a breeder is interested in crossing different species of plants to combine desirable characteristics to produce commercially superior varieties. So he found that I have two different plants with two different good things. I want to fuse them to get a hybrid plant out there. Okay. Now, what major approach in the crop improvement program would you suggest to the breeder to achieve this goal? and discuss in detail the techniques involved in this process. So this question appears to be from some other chapter, but it's from this chapter only. The answer, what is the approach in crop improvement program, is given in NCERT. Artificial hybridization is one of the major approaches of crop improvement program. So your answer is artificial hybridization, and I hope you remember how do you do it. Okay, the purpose is to cross different species, okay, or for forming superior commercial varieties, my dear children. The first thing that you do is, suppose it's a bisexual flower, it is having both the male and the female parts. So this is the male part and there will be the female part. So you remove the male part from this particular flower. This is known as emasculation. Only if it is bisexual flowers, that's when you do emasculation. That's the first step. Removal of anthers from the bisexual flowers. Why? Otherwise pollen grains will be landing on it. Again the same type of plants will be produced. We don't want that. And then we do one thing. After cutting that off, we bag this up. We cover this particular plant such that no other pollen grains can come and land, and such that we give some time for the stigma to be receptive. So that is known as bagging. So covering the flowers with butter paper to prevent unwanted pollen contamination. The next thing, pollination process. So once the stigma is receptive, once you know the stigma is receptive, what you're going to do is that you're going to open this bag, you're going to take the pollen from the plant that you like, the hybrid plants, the desired plant, my dear children. You take the pollen grain from that and you put it over here. You put it over here and then you do what? You do rebagging. You rebag it, you cover it again. That's the whole process, and then you wait for the progeny to be formed. Now for unisexual flowers, no emasculation is needed, only bagging before flower opens, that's all. So before the flower opens, you make sure you bag it, otherwise the pollen grains would come and land from other plants. We don't want that. We want the plant, the one that we desire, not all the plants out there. So your answer for this question would be artificial hybridization. And I hope you now know how to do it. Okay, now turn for the homework question which is out there, which is very important over here. So this is the homework question that you have two questions. Okay, pollen grains are well preserved as fossils because of the presence of sporopollen. As statement 1, sporopollen is the most resistant organic material known. Statement 2, no enzyme that degrades sporopollen is known so far. So you have all the statements. Tell me the answer in the comment section below. This is the 2024 question. The few flowering plants have evolved a special mechanism to produce seeds without fertilization. I explained you this. That's why I need the answer. So this is homework number 2 and this is homework number 3. Please put it down in the comment section. I want to see the answers. Now we would be going ahead with human reproduction, and from this point onwards I'll try to be a bit more faster, otherwise we won't be able to complete. Just give me a second's time. I will be back. Before that, if at all you want, you can pause the video and read the chapter. Okay, read the chapter, go through the summary of the chapter once, that would be enough. Okay, and then we will go ahead with the next chapter, that is human reproduction. And my dear children, if you think that this class is getting useful for you, you are able to revise things, definitely do not forget to click on the like button out there. Okay, let's get ahead. So 1 marker, 9 questions will be there, 2 and 3 marker, 5 questions, and 5 marker, there will be 4 questions in this particular chapter, that is human reproduction. Starting with the first question out there. Okay, statement 1 says the process of release of sperms from the seminiferous tubule is known as spermatogenesis. No, that is known as spermiogenesis. Is it? No, that is known as spermiation. I already told you, release of sperms from the seminiferous tubules, aviation, spermiation, that is the answer. Okay, spermiation would be the answer, not spermatogenesis. So this, it's wrong. Spermatogenesis means the formation of sperms. Now statement number 2, the spermatids are transformed into spermatozoa by the process called spermiogenesis. Is it? So they are saying there is spermatids and you are transforming into spermatozoa. Is it known as spermiogenesis? The answer is yes. So kids, okay, spermatids, which are kids, they are going to be mature into what? Spermatozoa. Okay, spermatozoa. So this is known as spermiogenesis. It is absolutely correct. So this is correct, but this is wrong. Statement 1 is wrong, and that is statement 1 is incorrect, but statement 2 is correct. Please make sure you're reading the question carefully before doing it. Okay. Now, the function of myometrium layer present in the uterine wall is 2. The uterus has 3 layers: perimetrium, myometrium, the middle layer, and endometrium, the inner glandular layer. What is the function of myometrium? To undergo contraction and relaxation, especially during what? During the process of parturition, when the baby is being moved out. So, to undergo cyclic changes? No, that is the role of what? Endometrium. Endometrium undergoes cyclic changes. Exhibit strong uterine contraction during parturition? Yes. Give protection to the uterus? That is the role of perimetrium, the outermost layer. Help in implantation process? That is again the function of endometrium. So, your only answer becomes B. Myometrium is for muscles. Myo is for muscles. What muscles? Smooth muscles. Are they under your control? No, it's under brain's control. It's involuntary muscle, my dear children. You cannot control it. The brain controls with the help of neuronal system or hormonal system. Okay. Now, going ahead. The hormone is secreted by ovary in the later phase of pregnancy. Later phase. So, ovarian hormone that we study, okay, is only one. During pregnancy, the ovarian hormone that we study is only one, and that is released during the later phase of pregnancy, towards the end of pregnancy, which is relaxin. What does the relaxin do? Relaxin says, cervical muscles, please relax. If you relax, then only you will have space for the baby to move out. That is a function of relaxin. This is not the answer, this is not the answer, this is also not the answer. So, ovary, relaxin, ovary, relaxin. So, ovary was working so hard, now it's asking others to relax. Now, during pregnancy, the fetus develops limbs and digits, my dear children. Very important question. So, always remember, first month, what do you get? After first month, heart. After second month, limbs and digits. After 3 months, you get all of this plus external genital organ and also organ system. Some organ system, external genital system, everything is formed by the end of the 3rd month, that is the 3rd trimester. 4th month is missed in your NCERT, 5th month is written. The baby wants what? Baby wants to move around. So, the first movement of the baby and also hair on its head. Okay, it says that I want to move and I want to show the style. So, hair on the head and the movement in the 5th month. By the 6th month, God says that you have hair, but on your body also. So, body hair. Okay, eyelids which are there, they separate. Eyelashes which are there, they also gets formed, and that's end. Okay, so by the 6th month, what do you call this as? 2nd trimester. You call this as 2nd trimester. This is what? The 1st trimester. So, during pregnancy, the fetus develops limbs and digits. But ma'am, month is not given over here. Weeks are given. No, in 1 month, how many weeks do you have? 4 weeks. In 1 month, 4 weeks. In 2 months, 2 into 4, 8 weeks. So, after 8 weeks, my dear children, you will find limbs and digits being formed. After 8 weeks, always remember that. So, first trimester, first 3 months. So, 3 into 4, 12 weeks can also be written. Similarly, second trimester means 6 into 4 would be what? 24 weeks. Always remember that. Next thing, in spermatogenesis, if FSH hormone is not secreted from the anterior pituitary gland, which stage is affected? In spermatogenesis, if FSH is not secreted from the anterior pituitary gland, which stage is affected? So, my dear children, I am going to make you listen to one story which is very, very important. Okay, so listen to me carefully. First thing, when men hits puberty, that's when hypothalamus secretes gonadotropin releasing hormones. This gonadotropin goes and acts on anterior pituitary. This anterior pituitary now releases 2 hormones, one is LH and the other one is FSH, follicle stimulating hormone. In the case of males, LH will be acting on the Leydig cells. The Leydig cells, where are the Leydig cells? So, if this is the testis and this is the seminiferous tubules, my dear children, over here, okay, the testis and seminiferous tubules cannot be like that. That would be wrong information, right? So, this is the testis, suppose. Over here you have testicular lobules. in the testicular lobules you have seminiferous tubules like this. Inside the seminiferous tubules, what do you have? You have Sertoli cells and you have the germ cells out there, which forms the sperms later on. In between the seminiferous tubules, you have what? Leydig cells. And what does Leydig cells do? Under the influence of LH, it gives rise to hormones like testosterone, male sex hormones. Now what will happen? FSH, my dear children, in turn will be acting on the Sertoli cells, which is there inside the seminiferous tubules, and now the Sertoli cells would be releasing some very important factors which ask the testosterone to please stay over here. So what is exactly happening is that from the Leydig cells, the hormones which is there, it will be going into the seminiferous tubules where the germ cells are forming the sperms. The germ cells which are there, they will be forming the sperms. And who is there to nourish? The Sertoli cells. And who comes inside the seminiferous tubule? Testosterone. Without testosterone, do you think this conversion is possible? No. So who says the testosterone to stay here itself? It's the Sertoli cells which releases some factors. And these factors are the one that says testosterone, please release, please stay over here. But who tells the Sertoli cells to release the factor? It would be the FSH. It would be the FSH, my dear children. So now the factors are released. Now what will happen? The process of spermatogenesis will be very, very smooth. Especially in spermatogenesis process, there is a last process which I told. It is known as spermiogenesis, where the spermatids are converted into spermatozoa. There the amount of testosterone is very, very crucial. So I say this particular factors helps in the whole spermatogenesis, definitely, but majorly in spermiogenesis. So formation of so in spermatogenesis, if FSH hormone is not secreted from the anterior pituitary gland, which stage is affected? Formation of primary spermatocyte, formation of spermatozoa, formation of spermatid, or formation of secondary spermatocyte? Your answer would be formation of spermatozoa. This would be affected. Spermatozoa is formed from spermatids. Spermatids transform into spermatozoa, right? So this particular step is being affected. So your answer is B. Next, to produce 2000 sperm and 400 ova, how many spermatogonia and oogonia are required? So when you talk about spermatogonia, my dear children, I hope you know that there is spermatogonia. This spermatogonia will be producing or differentiating. It differentiates into primary spermatocyte. This primary spermatocyte undergoes meiosis 1 and gives secondary spermatocyte. So it was 2n, now it has become nn. Here also it was 2n. First meiosis it undergoes and then it becomes half. Again meiosis it undergoes, you get what? Spermatids. They are all haploid in nature. And this spermatocytes, all of them, they form the spermatozoa. So how many? 1, 2, 3, 4. 4 spermatozoa or later sperms are being produced from one primary spermatocyte, or sometimes you also call that as spermatogonia. In this question, that is what they mean. So you have to find how many spermatogonia are required for forming 2000 sperms, my dear children. So one spermatogonia in general we say, or one primary spermatocyte in general, it gives rise to 4 sperms. So now you have 2000 sperms. So if you divide it by 4, you get 500. So can I just say that 500 spermatogonia are required? 500, each of them divides into 4. Okay, and that's how you get 2000 sperm. What about the ova? What about the ova? So first of all, again you need to understand there is oogonia. Oogonia differentiates, you get primary oocyte. The primary oocyte undergoes meiosis 1. It divides into 2, one cell and the other cell. One cell is the first polar body. It degenerates. This cell which is there, it is known as the secondary oocyte. The secondary oocyte again divides, meiosis 2. This forms the ovum. Okay, and over here it forms the second polar body, which is again of no use. So from one primary oocyte, you just get one ovum. Okay, from one primary oocyte, you just get one ovum, my dear children. So 400 ova is there. How many oogonia is there? Just say 400 oogonia is there, because only one can give rise to the one. Okay, Okay, so the answer would be 500 spermatogonia and 400 oogonia. Always remember, the number of sperm being produced is 4, the number of ova being produced is 1, the ratio would be 4:1. Cool. Jasmine, talk about the ratio. I told about the ratio. It would be what? 4 sperms and 1 ova, always. Okay. So the question can be twisted in the form of secondary. How many secondary spermatocytes are there? How many? In that case, please make sure you're reading to the question really carefully. Now, identify A and B in the schematic representation of hormonal control of spermatogenesis in humans. So let's decode it. Anyway, we have studied hypothalamus. Hypothalamus, my dear children, what does it produce? Gonadotropin releasing hormone. It will act on anterior pituitary, and then anterior pituitary will give LH and FSH. LH acts on Leydig cell. FSH acts on what? Sertoli cells. And Sertoli cells, my dear children, would be helping in the process of spermatogenesis. They help in what? They help in producing factors, and that factors help in the process of spermatogenesis, especially spermiogenesis. So your answer would be over here, gonadotropin releasing hormone and also Sertoli cells. D. Next question. X is a structure which gets embedded in the endometrial lining. The phenomenon is known as Y. What do you call that? X is a structure that gets embedded in the endometrium. What is that structure that goes into the endometrium and gets embedded? It is blastula. Blastula, or you call this stage as blastocyst stage also, where, my dear children, you will find an outer layer which is known as trophoblast, an inner layer of cells which is known as inner cell mass. This will further go ahead and form the actual embryo, and this will help in the formation of placenta. Well, this is known as blastula, and the process is known as implantation. Okay, it is known as implantation. So your answer would be D. Now, secondary oocyte released during ovulation. Just a second, okay? Released during ovulation can't be fertilized by sperm during what? Because, just a second. Secondary oocyte released during ovulation can't be fertilized by sperm because secondary oocyte which is released during ovulation, no? What happens is that there is follicles inside which the secondary oocyte is there, and it is released during ovulation. But still it cannot be fertilized by the sperm that easily. Why? Because secondary oocyte has not completed its division, it is stuck there. It does not form the ovum yet. Only ovum can fertilize with the sperm, right? So without ovum being formed, do you think fertilization can take place? No. So it is because it has not completed meiosis 2, not meiosis 1, it is meiosis 2. Once it completes that, that's when it will be fusing. Okay. Children, I'm actually getting a little bit tired, okay? So I'm so sorry if my energy level is drooping down a bit. Now, yes, question number 10. Parturition is induced by complex neuroendocrine mechanism. Comment. So let's get energetic, right? We have to be energetic. We have to do it. Remember, 100% effort from your end, 100% effort from my end. So let's go. So over here, we say that parturition, what do you call parturition? Means expelling of the baby outside the mother's womb, right? It's a complex neuroendocrine. That means nerves control and endocrinal control, both of them is there. Hormones are controlling and also the nervous system. How does it happen? So let's see that. So my dear children, suppose this is the uterus which is there, and here you have the cervix, right? So here you have the baby. When the baby is completely formed, it releases some androgens. Androgens are male hormones. Ma'am, females also produce it? Yes. And now over here, you have the myometrium layer, the muscular layer, and also the endometrium along with the placenta and things like that. So when the androgens are released, okay, the placenta will be converting this into estrogen. And what happens when the level of estrogen goes high at this particular point of time? There will be certain receptors that gets active. Where are these receptors, children? These receptors are in the muscular layer, that is myometrium. These receptors get active. What do you call these receptors as? Oxytocin receptors. That means they will start working when they receive oxytocin. So what has happened? Baby released androgen and the placenta converted into estrogen. Now the estrogen, when the level is high, it makes the oxytocin receptors really active. Where are these oxytocin receptors? In the myometrium. So all of this is happening, and one more very important thing is happening, that is the baby is trying to move out. When it tries to move out, my dear children, over here there are certain stress receptors in the cervix. Stress receptors that becomes very active, and they give the nervous information, the neural information through the nerves to the posterior pituitary of the mother. We studied about anterior pituitary until now, but we are now talking about posterior pituitary of the mother, not the baby, okay, of the mother. It gives a signal to the mother and says that, hey, posterior pituitary, you have to release oxytocin. Now this oxytocin will come and bind over here. When it comes and binds over here, there will be contraction, baby will try to move out. Again stress receptors become active, it will release more oxytocin will come here, again contraction. So this is, again, as you can see over here, hormones are involved. Yes, oxytocin is a hormone, estrogen is a hormone, all of them are hormones, and also nervous system is involved, my dear children, your neural system is involved. So this is known as, why it is known as that it is a neuroendocrine mechanism. So fetal ejection reflex, my dear children, this thing what the fetus does, this whole thing is known as fetal ejection reflex. Okay, so it is triggered by cervical stretching. When the fetus reaches the full term, its head pushes against the cervix, activating stretch receptors. Okay, not stretch, it is stress receptors. Now, nerve signals to brain. These stretched cervix sends signals to the hypothalamus. It will send signals to the hypothalamus. Now, the posterior pituitary secretes oxytocin in response. Now, strong uterine contraction, oxytocin stimulates powerful contractions of the uterus. Positive feedback loop. More contraction leads to more oxytocin release. So over here you are seeing that the signals are being sent, and oxytocin, which is the hormone which is being released. You don't need to write about estrogen and everything because that's not there in your NCERT. Okay, that's something that is out of your NCERT. So you just can write all this thing, that is certain stress receptors in the cervical region become active, and they would be giving signal to the brain, from the anterior pituitary, sorry, from the posterior pituitary, not anterior, posterior pituitary, oxytocin will be released, and now they will come and bind on the receptors in the myometrium, lead to contraction, and this will create a positive feedback loop, okay, until the birth has taken place. Now, in the figure given below, parts A and B show the level of hormones which influence the menstrual cycle. Study the figure and answer the questions that follows. So over here you can see certain hormones going up and down. This is A, this is B. In A comes FSH and LH, and in B comes estrogen and progesterone. Name the organs or glands which secrete the hormones represented in the label part A and B. So FSH and LH, my dear children, they are released from what? The pituitary gland. What pituitary? Anterior pituitary. And if we talk about estrogen and progesterone, my dear children, it is released from the ovary. Now, state the role of hormones secreted from part B on the uterus of human female during menstrual cycle. So B, part B. So you have estrogen and progesterone, right? So progesterone, what is its role during the proliferative phase? Let's talk about that. You have three different phases. Menstrual phase. Menstrual phase is when the level of progesterone, estrogen goes down. That's when endometrium is not maintained and falls away. Menstruation would be happening. And then you will have proliferative phase where the estrogen, my dear children, it will stimulate the endometrium growth and thickening little bit. It will stimulate that. It prepares the uterus for what? Implantation. Not necessarily, it's mostly the first point. Now in progesterone, what does it do during the secretory phase? In the next phase, that is known as secretory phase or the luteal phase, the level of progesterone increases and it stabilizes and maintains the thickened embryo. It makes it more thick and it also stabilizes and maintains it. Also one more thing happens, that is enhances the glandular secretion for the embryo development. So basically endometrium is like a gland, right? It is going to secrete a lot of things. So progesterone is the one that makes it secrete a lot of glycogen and things like that, such that if at all the embryo comes and attaches, it can get enough amount of nutrition. So those are the things that is being done by these hormones. So if fertilization does not occur, both the hormone level drops, leading to what? Menstruation. During what phase? The menstrual phase. So estrogen, it creates the basal layer of endometrium out there. Okay, it will help in the endometrial growth during the initial phase and the proliferative phase. Then progesterone, my dear children, will be stabilizing this, will be maintaining the endometrium and also enhances the secretions of the endometrium. Will that be okay to write? I hope it's okay to write. Okay, and I have taught taught you about a lot of things about this, what do you call, the hormones, or what is the exact way how the menstrual cycle works. If you have not watched it, please go and watch that video. Okay, in parts it is there in human reproduction. Please watch it. It's going to be really helpful for you guys. Okay. Now, during pregnancy, the level of hormones are increased several fold in the maternal blood. Name the hormones and write their importance. During pregnancy, the level of hormones are increased in several folds. So, my dear children, actually during pregnancy, there is formation of placenta. And placenta releases 4 very important things, that is HCG, human chorionic gonadotrophin, and there is HPL, and there is what, estrogen, and there is progesterone. So you might be thinking estrogen and progesterone are already released by the ovary, no? But here during pregnancy, the placenta takes up this job more seriously, and it does that. So we say 4 hormones are released by the placenta, but they are not asking about that. In your NCERT, there is a separate line given, that is, there are certain hormones whose level increases several folds. Okay, so HCG plus HPL are not usually produced, so they cannot be increased in several folds. You are talking about the existing hormones whose level increases. So what are those hormones whose level increases? One will be obviously estrogen and proestrogens, my dear children. Okay, so it is produced in large amounts by the placenta. It is essential for maintaining the pregnancy, for maintaining the endometrium, right? And now talking about the next thing, it is cortisol. It supports the metabolic changes in the mother. It assists in the fetal development. It helps in the development of the fetus. Prolactin, there is another hormone. Okay, prolactin. It prepares the mammary gland. It is there for lactation, so that the mammary gland will produce milk after what? After the process of parturition. Now, thyroxine, which is there, you know, thyroid hormone. Thyroxine, which is there, it is there for maintaining the normal basal metabolic rate, for improving the metabolism during what? The pregnancy. So these are the hormones that you need to write down: cortisol, prolactin, thyroxine, estrogen and proestrogens also. Cool? Will you be able to write it? Will you be able to explain it? I hope you do. Well, if you're confused what is progesterone and progesterone, my dear children, so basically I can write progestogen. It can be of 2 types, natural and synthetic. So the natural one which is there, you call this as progesterone. You call this as progesterone. It's the same thing only. Okay. So cortisol, prolactin and thyroxine are the one, and all this thing are the one which are increased in several folds. They were there before, they are increased now. And these hormones, if you say, especially HCG and HPL, they are only produced during pregnancy itself. Okay. So to test pregnancy, if at all someone is pregnant, what do? You do HCG test. Okay, in the urine test, you see the pink line, everything is found because of the HCG itself. Now, list the functions of human placenta. What is the function of human placenta? It is there for nutrient and waste exchange. So, you have to tell the good part of the story and then the bad part. It supplies oxygen and nutrients to the embryo, and then what does it do? It removes carbon dioxide and excretory waste materials produced by the embryo. That is also there. Now, endocrine function, there is one more function. So, apart from nutrient and waste exchange, there is an endocrine function that it does. It acts as temporary endocrine tissue. It acts as what? Temporary endocrine tissue. Okay, it is not always going to produce hormone, and it is not just, no, it's not like that. Basically, it's not going to be there forever. Placenta will be only there until the baby is born. Right, so it is going to act like a temporary endocrine tissue which is there. It produces important pregnancy related hormones such as HCG, human chorionic gonadotrophin, and HPL, human placental lactogen, and estrogens and progestrogens. Please remember, all of them are very important. HPL, human placental lactogen, as the term suggests, lactogen, it is helping in lactation for the formation of mammary gland, for the proper thing, okay, for proper lactation to happen. HCG, human chorionic gonadotrophin, what does it do? So my dear children, what usually happens is that, I hope you know the menstrual phase, in the menstrual cycle there is ovulatory phase, where what happens? The Graafian follicle ruptures, and what is released? The secondary oocyte is released, and it waits for the sperm to come and attach. But what happens to the ruptured Graafian follicle? It will form something that is known as corpus luteum. First of all, why did it rupture? Because the amount of LH was way too much, and also FSH, but it is majorly LH whose amount is way too much. right? So we say LH is the one that maintains the corpus luteum as well. It broke down the Graafian follicle, but later it maintains the corpus luteum. This corpus luteum in turn basically it produces progesterone and estrogen, and progesterone is the one that maintains what? Endometrium. So if you want pregnancy to happen, you need endometrium and you want progesterone to be there. But my dear children, there is a twist. When the level of progesterone increases, it says to the hypothalamus to stop the secretion of these guys. But then it does not realize that if these guys are not there, corpus luteum will not be there. If corpus luteum will not be there, it itself would not be there, my dear children. So it usually happens like that only. If at all fertilization has not taken place, this guy tells the other guy and then the whole thing goes off. It will take some time to go off, but it goes off. The level of progesterone will be decreasing, and that's where menstruation and things like that will be happening. Now we don't want that to happen. We want someone to maintain the corpus luteum. So during pregnancy, my dear children, it is HCG that takes up this role. HCG actually maintains the corpus luteum such that the level of progesterone never decreases. Get it? So that's the role of HCG. So nutrient and waste exchange and also endocrine function, these are the functions of human placenta. Now briefly explain the sex determination in human being. So humans have 23 pair of chromosomes. I hope every one of you agree. We have 23 pairs of chromosome. Half of it, okay, we receive from one parent, half of it we receive from another parent. That is 23 number of chromosomes from one parent, 23 number of chromosomes from another parent, 23 plus 23, 46 number of chromosomes. But if you talk about pair, 23 pair only. In each pair there are 2 chromosomes, no? So 23 pair. Now my dear children, out of them 22 pair of them are autosomes. Autosomes means they have nothing to do with the sex of the organism. They are the chromosomes which carry the genes coding for all the other features of us, but not coding for the sex. Now one pair, the last pair, the 23rd pair is the sex chromosome out there. Always remember the number of autosomes is 22. A number of sex chromosome is just one pair, that is the 23rd pair. 23rd pair, my dear children, that is the sex chromosome pair. Okay, there are 2 chromosomes, only 2 chromosomes. So in the case of females, you have X and X chromosome. Here you have X and Y chromosome in the case of males. So if you have one Y chromosome, that means you are male. Now if you are asked about gametes, so gametes can have either this chromosome or that chromosome. Either way, it will be X only. Whatever you do, it will be X only in the case of females. In the case of males, it can be either X or either Y. Now, when the gametes come together, this and this gamete can come together forming XX. This and this can come together forming XY. This and this comes together forming XX. And this can together forming XY. Okay, so there are, if you see, this is a girl baby, this is a girl baby, this is a boy baby, this is a boy baby. So 50% of chance are there that a girl can be born and 50% of chances there that a boy can be born. But who decided it can be girl or boy? It's basically the sex chromosomes of the males, because they are 2 different types. They are of 2 different types, X and Y. Also remember one very important thing, that Y chromosome is small in size. The Y chromosome is small in size. Okay, there's a structural difference also. So my dear children, out of 23 pair of chromosome, 22 pair, my dear children, are of autosomes. The last pair, the 23rd pair, which is there, it is the sex chromosome. It can be either XX or it can be XY. If it is XY, it's a male. If it is XX, it is a female. Why? And who decides the sex of the child? It is the male. And it's actually in the class also which I took, I told you it's not correct to say like that, but that's how, if you took genetically, it's like that. Okay, so please remember that and please write your answer properly like this. Fine, you will get the PPT, do not be worried. Now 5-marker question where you have to draw a lot of diagrams. So draw a labeled diagrammatic sectional view of the human female reproductive system. In that case, you have to draw like this. Okay, so you would have the uterus, and then you would have the fallopian tube. Yeah. So I cannot make it here, I will make it in the next slide. So my dear children, this is the uterine fundus. So from here to here, you have finger-like projections. Okay. Okay. So here what do you have? You have the ovary. Similarly here what do you have? You have the ovary. Draw it properly. I'm just giving a rough idea. Okay. So this is the ovary that you are having. This is what? The fimbriae, the finger-like projection that you are having. This is what? Infundibulum. Further, you will have a broad ampulla and then you have a narrow isthmus. This is known as, this whole thing is uterus, so this is known as uterine fundus. This is your uterus, and over here you have the what do you call the perimetrium, and then you have the myometrium, my dear children. You also have the endometrium. Okay, you have the endometrium. Now when you reach over here, you would be having the cervix. This would be the cervix, and then you would be having the vagina. Fine. This is the cervix, this is the cervical canal, and this would be the vagina out there. Where does fertilization happen? Over here. The egg waits over here. Okay, so this is a general thing. Please write the names: perimetrium, myometrium, endometrium. So the picture, it looks actually like this. You can follow this particular diagram. It will be easy for you to remember. Please make sure you're writing it. Okay, and please make sure you're drawing it and seeing it. Do not learn the picture. You cannot learn a picture. You have to draw and do that. Similarly, draw a neat label diagram or sectional view of mammary gland. Again, this is also comparatively really easy. First thing, my dear children, always remember that over here, I'm just going to draw this part. Okay. Should I draw in white or black? I'll draw in white. So there are lobes. You can just imagine certain lobes like that. Okay, there are almost so many lobes, 15 to 20 lobes. Anyways, so over here you have alveoli. So many of them. Alveoli which is there, it will be opening up into tubules. Into what? Tubules. Tubules will be joining together. Tubules would be joining together to form what? Duct. Now the duct which is there, it will be coming to join together to form. So this duct, and suppose there is another duct, it also joins over here, and it will form the ampulla. And the ampulla later forms the ampullas, all the ampullas. Okay, so here another ampulla will be there, another ampulla will be there, another ampulla will be there. All of them, my dear children, all of them will be opening up together into what? The lactiferous duct out there. So first thing, you have lobes. The first thing, you have lobes, then you have alveoli, then you have tubules, and then you have duct, then you have ampulla. All the ampulla would be coming up together like this and opening into lactiferous duct. Okay, so this thing you have to practice, then only you will be able to do that. Over here they have not mentioned the lobes separately, so you don't have to draw the lobes like this. But in general, just for an idea I told you, okay, for an idea. So here they have not even specified how does it exactly look, but yes, this is how the thing works, and there will be fat all around over here. This particular side, there will be fat. I have not made a proper diagram, as I told you. This is just for your reference. You would have to draw and practice it, otherwise you won't be able to remember. So only if you remember the pattern, okay, that is MAT, mammary lobes, alveoli and tubules, then DAL, duct, ampulla and lactiferous ducts. MAT and DAL, if you remember, you will be able to draw it also. Fine. So this is a repeated question, it usually comes, make sure you draw it. Now draw a neatly labeled diagrammatic sectional view of male reproductive system. My dear children, male reproductive system, all the parts are important. Jasmine talk about 2025 question about male accessory glands. They asked what are the accessory glands involved in the case of males. There are 3 accessory glands. What are those? Seminal vesicle, prostate gland, bulbourethral gland. Out of them, how many of them are paired? Seminal vesicle and bulbourethral are paired. There are one pair of it. Okay, prostate gland is not paired. You need to know the secretion. Seminal vesicle, it secretes what? It secretes fructose, not sucrose, not glucose, fructose. Okay, what does prostate gland secrete? Calcium and also enzymes and calcium, clotting enzymes and calcium. Bulbourethral gland is responsible for lubrication. What do you call all of the secretion? Seminal plasma. All of it you have to recollect and remember. What are the accessory ducts out there? Remember the accessory ducts. So over here again a picture. Children, do I have time? Let's see. Okay, I'll try to do it. The accessory ducts which are there. So we'll start with the what do you call? Okay, so this is the what? This would be the urinary bladder of the male, right? So urinary bladder comes from the top. You have all the other things. So this is the ureter which is there. Fine. And the urinary bladder would be going up like this until here. This opening which is there, it is known as urethral meatus. This is the urinary bladder. Okay. So over here, the thing that you need to know is, first of all, you have the testis. In the testis, my dear children, the important thing that you need to know is there are testicular lobules. Okay, if I draw like that, I won't be able to make the other things. Okay, there are testicular lobules. Inside the testicular lobules, you have seminiferous tubules, right? The seminiferous tubules will be opening up and coming over here and forming a structure like this. This is known as rete testis, the first accessory duct according to your NCERT. The rete testis will be coming up, moving out of the testis like this with the help of the next thing, which is vasa efferentia. The vasa efferentia forms a big long thing out there, which is known as epididymis. Now the epididymis will be going towards the uterus with this particular thing which is known as vas deferens. So all of it that you see over here from 1, 2, 3, 4, they are all accessory ducts. They are all accessory ducts. Now they have reached over here. From here, my dear children, so they come over here. So here also, from here also, just imagine one is coming like this. Now they will get a secretion from a very important thing that is known as seminal vesicle, as I told you, the first accessory gland. And now both of them together will form something like this, which is known as the ejaculatory duct. Ejaculatory duct. Ejaculatory duct will be opening into the urethra. It will be opening into the urethra. Now this part is covered by another gland which is out there, which is unpaired gland. This gland is known as prostate gland. Okay, so ejaculatory duct is also a part of what? The male accessory duct which is there. Then it joins to the urethra. This is what? The urethra through which the urine passes. Okay. Now over here, my dear children, you have the external genital organ, that is the penis. At the top of it you have the next gland. This is first gland, seminal vesicle. This is second gland, that is prostate, and this would be Cowper's gland or bulbourethral gland. And with that we finish the male accessory duct. Same thing, you have to draw it there also properly. This is, I drew it big because I wanted to make you understand this particular thing. Okay, please draw like that and you get your marks. Now write the schematic representation of spermatogenesis. Write the two events that occur in the ovary and uterus during follicular phase of menstrual cycle. So first we'll go with the next question. Okay, what happens in the ovary and the uterus during the follicular phase of menstrual cycle, my dear children? Follicular phase means follicles are developing. So obviously in the ovary you have the follicles. So the primary follicles in the ovary are growing into what? Fully matured Graafian follicle. Okay, now in the uterus what is happening? The endometrium of the uterus regenerates through proliferation. What is happening? The endometrium just fell down, right? Now it is going to reform again. The endometrium of the uterus regenerates, my dear children. That happens in the uterus during the follicular phase. Okay, under the influence of estrogen. There is no progesterone as of now. It is estrogen that will be helping in what do you call proliferating the endometrium. Where do we get the estrogen from? So basically, when the follicles are developing into graphene follicles, no, the follicles releases some amount of estrogen. And this estrogen is helping in the maintenance of the endometrium. It is helping in the formation of a bit of endometrium during the follicular phase itself. Now talking about spermatogenesis, we already spoke about that. There is spermatogonia, which later forms the primary spermatocyte by just mitosis and differentiation, nothing else. Does not undergo meiosis. The primary spermatocyte, which is having 46 number of chromosome, undergoes, primary spermatocyte which is having 46 number of chromosome undergoes division to form secondary spermatocyte, my dear children. The secondary spermatocyte again divides, you get spermatids. The spermatids transform into what? Spermatozoa. So that is how the whole process works. I already explained you that. Don't think we need a newer explanation. Okay. Do we need it? No, right? I hope no. My dear children, if at all you have any doubts still persisting, you can put it down in the comment section. I am looking at the time and getting scared because I know if it's too long, you also would not be very much, comfortable to watch it. So that's the reason I'm trying to go a bit fast, but I'm trying to cover a lot, which is why it's taking a lot of time. Now, which of the following is essential for maintenance of endometrium and for implantation of fertilized ovum? This is your homework question. Estrogen, progesterone, LH-FSH or androgen? This is homework number 4. Tell me in the comment section. Now, we are going ahead with the next chapter, which is reproductive health. And over here we have, I have not filled in how many questions are there. 15 1-mark questions are there. I think we would have to skip a few of them. So whenever I see a question, I will be skipping a few, okay? So select the one among the given that is probable reason for explosive growth of population in India. This came in 2025. So what is the reason for population growth? A rapid increase in MMR. MMR means maternal mortality rate. Mothers are dying. If that rate is increasing, that means it's not good. Okay, obviously this is not a good reason for population increase. Population decrease would be happening. A rapid increase in infant mortality rate, IMR, that is also wrong. If infants are dying, my dear children, how can you expect the population to increase? Increase the number of people in reproducible age. Yes, if number of people in reproducible age are increasing, then obviously the population is also increasing. Okay, the rapid increase in death rate, wrong. A rapid increase in death rate would be wrong. That won't increase the population size. Now, which one of the following ART can be used? Artificial reproductive technologies can be used, assisted reproductive technologies can be used to rectify the infertility due to low sperm count. Low sperm count means you need insemination, artificial insemination, right? Because the sperms are not enough for the whole process to happen. So you have to take a lot of sperms out there and then artificially you have to do it. Now, among them, what is the artificial insemination technique? Gift? No. IUT? No. IUT means intrauterine transfer. What are you transferring? You are not transferring sperm. You are transferring embryo. Okay. ZIFT? No. IUI? Yes. Intrauterine insemination. This is your answer. When low sperm count, go for intrauterine insemination. Next question. Out of the options given below, choose the correct stage for transfer of the fallopian tube for successful IVF results. If you want to transfer something into the fallopian tube, it can be either a zygote or an embryo, but only up to 8 blastomeres, not beyond that. If it is beyond 8 blastomeres, it has to be transferred to uterus, and that is known as intrauterine transfer. If you are transferring to the fallopian tube, it is known as ZIFT. Zygote intrafallopian transfer. It's not just for zygote, it is also for the embryo, which is up to 8 blastomeres. Anywhere if fallopian tube is mentioned, then up to 8 blastomeres only, not beyond that. Okay. Now, sperms produced by seminiferous tubules are transported through accessory ducts. Which duct should be tied and cut for male sterilization? If you do not want the sperms to be going to the semen, be a part of the semen, my dear children, what is that something that you are going to cut? You would be cutting vas deferens. You would be cutting vas deferens. Vas deferens is the one that takes things to distance. You can think like that. Not vasa efferentia, not retitestis, epididymis. One important question. In human beings who undergo vasectomy, that is this particular procedure, do the semen contain the sperm? The answer is no. The semen does not contain the sperm. The semen just contains the seminal plasma that is secreted by the accessory gland. Sperms are missing. But are the sperms being formed in their body? The answer is yes, but they cannot be transported. Spermatogenesis happens, but sperms are not transported. Remember that. In the case of females, if fallopian tube is cut, is menstruation gonna happen? Yes. Why? Because ovulation is still happening. Ovum is still being released, but ovum is not getting transported. So it will degenerate, but it is being produced, no? So all of it is normal. Just that they are not getting transported. Which of the following sexually transmitted diseases is not completely curable? You have three of them. Three of them has a H in it. Genital herpes, hepatitis B, HIV AIDS. Three of them are non-curable. Rest all of them are. So over here you have genital herpes and this becomes your answer. Next question. An example for non-medicated IUD, intrauterine device. Intrauterine devices are methods of contraception. Okay. And which one is non-medicated? The one that does not release anything. Okay, but still inhibits all the process. It is lipase loop. It is what? Lipase loop. I taught you this particular thing. Please go and watch the video if you have not seen it. It looks like this. It is put inside the uterus. It makes what? The condition hostile. For example, it says that I am foreign, please attack. When our body becomes in the attacking mode, whatever comes into our body, that will be killed along with the sperms. Okay, the sperms are also being killed. So lipase loop is an example of non-medicated IUD. Now, an assisted reproductive technology in which a sperm is directly injected into the ovum. If sperm is directly injected into the ovum, you call this as intracytoplasmic sperm injection, ICSI. Inside the cytoplasm of what? The egg. The sperm is being injected, ICSI. Now, menstrual cycle does not occur during intense lactation. It is due to. Why does menstruation stop during intense lactation? This is known as lactational amenorrhea. A method of natural contraception, not conception. Why? Because the mother who is lactating in her body, prolactin hormone will be way too much. Prolactin hormone is way too much. Now, when prolactin is way too much, it does not allow FSH and LH to increase. And if FSH and LH is not there, follicles can develop into mature follicles? No. If no mature follicle, ovulation can happen? No. So, inhibition of LH is a reason. It can be FSH or LH, but over here the first point itself is correct. Now, coitus must be avoided from day 10 to day 17 of menstrual cycle. The reason is, why is day 10 to 17 important? Because it is known as the fertile period. Our cycle is for 28 days, my dear children. On the 14th day, we expect ovulation to happen. But 4 days before it and also 3 days after, it's not safe. Why? Because ovulation can be shifted anywhere. That is why 10 to 17 days, which is the fertile period, during this, sex should be avoided. So, sperms remain viable for 7 days. That is why it must be avoided. No. Ovum may be available for fertilization. That is the reason. Ovulation can be happening. Ovum might be available for fertilization, which is why it should be avoided. Okay, just give me a second. Okay. Going ahead with the next thing. Condoms have additional benefit as compared to other contraceptives because. Why does condom have additional benefit as compared to other contraceptives? Because, my dear children, condoms does not allow genital fluids to get mixed up. Okay, so it prevents what? Sexually transmitted infections, which are also transmitted through what do you call? The fluids. Okay, and going ahead, just a second. Okay, so there is statutory ban on amniocentesis as it reveals the chromosomal anomalies. It can be used for determining the sex of the unborn. It is used to reverse the sex of the fetus. It can be used for correcting genetic disorders of the fetus. The answer is, why do you have to put a ban? Actually, amniocentesis is used to find the genetic abnormalities, my dear children. But it's good to find the genetic abnormalities, right? What bad was done such that a ban came, my dear children, because people were using it for the determination of what? The sex of the unborn. So this becomes the wrong answer. Okay, now going ahead with the next thing, select the option which with correct identification of the given contraceptive measure. So IUD, CUT, subcutaneous implant, progesterone or estrogen combination, my dear children. So what is this? This is actually an implant which you put under the skin and it releases progesterone or it produces progesterone estrogen combination, my dear children, both of which have an inhibiting effect. So when the level in the body is normal, it's fine. But when we are giving it extra, it becomes totally abrupted. Okay, so your answer would be C. Next question. a lady is 8 weeks pregnant, various tests done confirm that if her child is born, it would suffer from serious illness. What is the best suggested option in this case? If the child, various tests has been done and it is clear that the baby would be born with serious illness, what can be done if she is 8 weeks pregnant? My dear children, MTP can be performed, medical termination of pregnancy can be performed. It is safe up to 12 weeks. It can be done up to 24 weeks, but it is safe? No. It is safe only up to 12 weeks, but remember it can be done. If the question is how, until what time it can be done? Until 24 weeks. That is the second trimester. You can do it, but it is preferably safe to do it here itself. Now going ahead with the next thing, if a female has rudimentary ovary but can provide a suitable environment for the growth of fetus, the art technique that can be used is, if rudimentary ovary, that means the ovary is not producing ovum, but the uterus is totally fine, then what can be done? Gift can be done, that is gamete intrafallopian transfer. You can give the gametes and that can be put into the fallopian tube. She cannot produce the gametes, that's the problem, no? So you give the gametes, so that is known as gift. It is usually, it is done only with the females, okay? Now, just read out the question. I cannot explain as of now. As you see, the time is running up. Mention the 2 medical grounds on which the pregnancy is subjected to termination, when it is risk to mother's health, when it is risk to fetal abnormality as well. Please write all of this thing. Medical Termination Act was put down in the year 2017. When was MTP legalized? In the year 1971. Ulta of this, 1971, here 2017, it was Medical Termination Act was put down and it was said that it is done in the case of 2 conditions. And I have taught you in the chapter that, for example, if a person has going to undergo abortion within the 3 months, then you need a medical practitioner's requirement out there, okay? Otherwise, you need approval of 2 medical practitioners if you're going ahead with the second trimester abortion. Now, what are emergency contraceptives? Right there, hormonal combination, very important question. Emergency contraceptives are pills that is used to prevent pregnancy after unprotected sexual intercourse or contraceptive failure. The use of it is to what? Within a short window. It has to be used within 72 hours. Remember, 72 hours, not 1 week. Within 72 hours, it has to be taken up. And basically over here, you're given progestrogens or progesterone-estrogen combination. Or you can also give IUD. Okay, IUD also can be taken up over. IUD can also be used as what? Emergency contraceptive, my dear children. Not hormonal IUD, you have copper T. That is something that can be used. But within 72 hours, this has to be done. Either you give pills or whatever has to be done, it has to be done within 72 hours. Okay, so usually the hormones which are there, they prevent ovulation, that does not allow implantation to happen, and they do not allow the sperms to enter. Well, the sperms have already entered now, but at least ovulation and implantation can be prevented with the help of these hormones out there. Now, what is infertility? Infertility is basically when someone have a sexual intercourse, unprotected sexual intercourse, but still do not have children. The reasons would be these. Please go through all the reasons if you want. You can go through the PPT once and read them. What are the features of ideal contraceptive? User-friendly, easily available, effective. It should be reversible. Minimal no side effects, should not interfere with the sexual drive or activity of the organism. Now, the popular and effective contraceptive include IUD. Mention the types of IUD with example of each. So there is non-medicated IUD which does not release anything but just makes the uterus hostile. Okay, it says that I am foreign, please make more and more WBC such that anything that comes along with me can be killed. So they are simple inert devices that prevent sperm from reaching the egg by creating a physical barrier in the uterus. By creating a physical barrier and also by activating what? By activating the immune system. This is the major point. Immune system. Second is copper releasing IUD. Copper has 2 functions. One is to decrease the sperm motility. Second is to decrease the the sperm's fertilizing capacity. Sperm motility decrease, sperm fertilizing capacity also decrease. Example, copper T, or you have copper 7 which is in 7 in shape, or multi-load 375. 3 examples are there. Hormone releasing IUD, 2 of them, LNG20 or progestasert. Over here basically these IUDs are going to release hormones such as progestrogens, and these hormones when they are there, they do not allow implantation to happen, they do not allow ovulation to happen, and the cervix which is there, it will be having thick mucus such that the sperm would not be able to enter. 3 things. Okay, so please go through them. It is really important for you to read through. Going ahead, suggest 3 simple principles through which we can prevent sexually transmitted diseases. So please practice safe sex, Maintain monogamous relations, as in avoid having multiple partners. And please go to qualified doctors if at all you feel that there are certain symptoms that is there in your body. Okay, and there are certain important symptoms given in your NCERT. Please go through them as well. It has not been asked yet, but it can ever be asked. We never know, right? What is contraception? Briefly explain 2 barrier methods and 2 surgical methods. Surgical methods, vasectomy and tubectomy. Vasectomy, you cut the vas deferens. Tubectomy, you cut the fallopian tube. And now natural method, there is periodic abstinence where you do not have coitus during 10 to 17 days of periods, 17 days of your menstrual cycle. Now withdrawal method or coitus interruptus is where you withdraw the penis before ejaculation takes place. And then is lactational amenorrhea, which is also one method. Please go through all of this thing. If you want, I can move aside, but anyway you will get the PPT, so please do not be worried. Okay. And last question, homework question, which one among the following is contraceptive method that involves no medicine or device? No medicine or device. Condom, cervical caps, Haley, coitus interruptus. This would be your 5th homework question. Make sure you're doing it. This is from the year 2000, not 2024, 2025. Okay, cool. Now we are going ahead with the most important chapter out there. Okay, that is principles of inheritance and variation, and I'll be trying to teach you some important concepts as well. Okay, so 1 mark question there are 8, 2 mark question there are 5, and 5 mark question there are 3 of them. Let's begin. So 37.2% recombinant Drosophila progeny obtained in the T.H. Morgan's Dihybrid cross experiment with the phenotypes red eye color, normal body, and white eye color, miniature body is due to. So basically this question says, is asking you one very important thing. If I say there is 37.2% recombination, then 37.2% recombination, that means there is enough amount of recombination. New combinations are being formed. Okay, combination is in what sense? So basically you're talking about these things. So that is what? White eye color and miniature body. White eye color and miniature body. Now over here, my dear children, just understand one very, very important thing. That is, suppose over here I am removing all this thing and I am writing something else so that you understand it better. This is capital A, this is capital B, this is small a, and this is small b. Okay, here these genes which are there, they are very far away from each other. You say that they are loosely linked. They are loosely linked, or they have so much distance between them. You say you have a longer distance between them. Longer distance between them. Now suppose there is another scenario which is like this. Okay, here you say that they are tightly linked. Tightly linked, okay, or you say that they have shorter distance. So basically, what's the use of this? And this is that. Suppose later all the chromosomes which are there, they come in phases where they exchange material. Where they exchange material, my dear children. So in this phase when they exchange material, it can happen like this. Capital A can remain over here, but this part can come over here, so it will be small b. And the other part which is there, it can go there, that is capital B. And over here, small a can remain the same. If you see now, there is a new combination. There is capital A, capital B over here, but here there is capital A, small b. Okay. Why is there a new combination? Because these genes which were there, they were loosely linked. That is why you find new combination. And when you say new combination, can I say recombination? This is what? A new combination, recombination. You are recombining something else. But over here, if you try the same, what will happen? If this parts go that side, it will be capital A, capital B together. Here, these genes would be still together. Why? If I talk about recombination here, is recombination actually happening very much over here? The answer would be no. Very less amount of recombination. Why? Because these genes are very closely linked or very tightly linked. So in in the experiment which was done by T.H. Morgan, he said 37.2% recombination was seen. if I say that 50% is the highest amount of recombination anything can ever achieve, my dear children, if it has reached 37.2% of recombination, then don't you think the genes that he is talking about for white eye color, miniature body, or red eye color and normal body, my dear children, these genes are loosely linked. They are far away from each other, which is why you find new combinations out there, recombination out there, that is 37.2. If in the question it was said that 1.7% recombination is there of these particular genes, your answer would be that these genes are tightly located, tightly linked to each other, or they have shorter distance. But in this case, where the recombination is more, we can assume that they are loosely linked with each other. So your answer for this question would be loosely linked and shorter distance? No, loosely linked and what? Longer distance. Loosely linked and longer distance. So always remember this particular thing. You have your best friend with you. You both are very close to each other. Do you think someone else can come here? No. So you guys are tightly linked and you have very shorter distance. But now suppose I have a friend and there is another friend, my dear children. So now this combination is what? Loosely linked. There is longer distance and now new combination. So it's not like that. Remember. So first scenario is you and your best friend. If you are very close to each other, there is no chance that a third person can come in. You guys are tightly linked, a shorter distance is there. But now, if you and your best friend are not really close, what will happen? A third person can come over here. There's new combination that can happen. So that is loosely linked and longer distance. I hope you get it. Okay, so that was told by T.H. Morgan. He was explaining that, my dear children, there is something that is known as linkage and recombination. Over here, they are loosely linked, and loosely linked ones will always show more amount of recombination. That is what he was talking about. Now, going ahead with the next thing. In a dihybrid cross in pea plants, Mendel got 9:3:3:1 phenotypic ratio. It denotes that. What does it denote? So for explaining you this question, I have to go to the last question, which is actually good because I would be able to explain you the important things that I have not explained you until now. So my dear children, when do you get a 9:3:3:1 ratio? First of all, see over here. Okay. So suppose you have round and yellow and wrinkled and green plant. So usually when you talk about monohybrid cross, you talk about tall and dwarf and things like that. You just have one character, that is height. But over here you're dealing with two characters. One is the shape and one is the color. So for round, it is capital R, but you need two genes for that. Similarly for yellow, it is capital Y, capital Y. Over here you have taken pure wrinkled and pure green. So it will be smaller, and here it will be small y. Because yellow is dominant, the smaller version of it would be y. So in pictorial representation, it looks like this, my dear children. So you have chromosome capital R, and you have chromosome capital Y. Similarly over here, you have chromosome small r, and you have chromosome small y. Now, when you form the gametes, my dear children, what are the combination? Whatever you do, from this pair of chromosome, one can come in. So it will be obviously always capital R. From this pair of chromosome, it will be obviously only capital Y that can come in. Either way, whatever you do. So you can get four combinations, this and this, this and this, this and this, this and this. But anyway, it will be capital R and capital Y. Okay, capital R and capital Y. Similarly over here also, whatever you do, it will be small r and small y in the gametes. So in the terms of chromosome, your thing would be looking like this. Capital R and capital Y. Here it will be looking like this. Small r and small y. Agreed? Now suppose this and this gametes comes together. What will happen? You will get capital R, capital Y, small y. Agreed? Capital R, capital Y, small y. How does it look? So there is capital R, there is small r, there is capital Y, and there is small y. So this was the F1 generation of Mendel. All the plants that he saw were round and yellow. Were what? Round and yellow. But again the confusion came. Is it round and yellow like this? Or is it round and yellow like this? We never know. Or is it round and yellow like this. It can, so many combination can be possible, my dear children. So what did he do? He did self-crossing, just like he did previously. So for self-crossing again, my dear children, this is F1 generation that I got. Now I am going to cross the F1 generation, okay? Capital R, small r, capital Y, small y. Capital R, small r, capital Y, small y. Okay, if you, as I told you, it looks like this. It looks like this. Now, my dear children, this can go with this. This particular chromosome which is there, it can go with this particular chromosome. One guy has to come from here, one guy has to come from here. So this capital R and capital Y can come. Capital R can come with small y also. Agreed? Small r can come with capital Y. Small r can come with small y. Agreed? Similarly, there in the terms of gametes, here also, my dear children, 4 gametes were formed, but all of them were same, which is why we wrote it once. So here also the same things is going to be happening. Capital R, capital Y, capital R, small y, capital R, capital R, small y, small r, capital Y, and small r, small y. These are the gametes. Now, my dear children, you have to fuse them. Now, you cannot keep on crossing that, it will be too much of work for you. So, what do? You make the Punnett square. Okay, if I remove it, everything will be going. So, I can still try to make it over here itself. Suppose I make it here and I show you. Okay, so I am removing this from this side and writing it over here. Capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. Suppose this and this comes together, I get capital R, capital R, capital Y, capital Y. This and this comes together, I get capital R, capital Y, small y. This and this comes together, I get capital R, small r, capital Y, capital Y. This and this comes together, I get capital R, small r, capital Y, small y. So in this way, I get so many combinations, my dear children. I get how many combinations? I get 16 combinations. I get 16 combinations. And over here, 9 of them, the ratio that comes at the end would be like this. 9:3:3:1, where 9 of them would be the dominant character, that is round and yellow. 1 would be the recessive character, that is wrinkled and green. Wrinkled and green. 3 of them would be new combination. Here 3 would be, round was with yellow, right? Now this time it will be round with green. Over here wrinkled was with green, right? Now wrinkled will be with yellow. So what do you understand from this experiment that he did? That in dihybrid cross, my dear children, the genes for one particular trait are assorting independently for the genes of another trait. So this was for shape and this was for what? Color. Do you think they are getting mixed anywhere? No, they are. Initially we studied that both the chromosomes and the factors are segregating in the law of segregation, right? In the law of segregation we studied that whenever gametes are being formed, the chromosomes or whatever is there, the factors would be separating and each gamete will be getting a factor each, right? Over here we are studying one another thing, that is when you talk about traits, okay, the alleles for different traits. The alleles for different traits are going to assort independently. They are going to move around independently. If they were not moving around independently, my dear children, you would not be able to see these many new combinations out there. You will always see round with yellow only and you will always see wrinkled with green, but that's not the case. Wrinkled is with yellow and round is with green also. That's because of law of independent assortment that Mendel put forward. He said that the gametes that you see, the alleles that you see, they are going to segregate independently because they are belonging to different traits. Okay, I hope this is clear. Fine. So always remember 9 for the dominant trait, 1 for this recessive trait, 3 for the new one, again 3 for the new combination that is being formed. Please take a screenshot if at all you need it. Okay, now I am going to show you over here. So write the schematic representation of explain dihybrid cross. This is how you have to explain. Capital R, this thing, the same thing that we did. They made the Punnett square, they have shaded the color also so that you can understand it better, my dear children. Now law of independent assortment. When 2 pairs of traits are combined in a hybrid, segregation of one pair of character is independent of the other pair of character. Segregation of one pair of character. Okay, one pair of character as in you are talking about capital R, small r. This will segregate independently of the other pair of character, which is what? Capital Y, small y. There's this law of independent assortment. It will take some time to sink in, but it will. Cool? So, when two pairs of traits are combined in a hybrid, one pair of character segregation would be happening independently of other pair of the character. That is law of independent assortment. Okay, and it was only got after the dihybrid cross. Remember over here, capital R, capital R, capital Y is round yellow. This is also round yellow. This is also round yellow. Why? Because over here you just need one dominant trait. Over here you just need one dominant trait. Always remember that. Okay? And even this is round, yeah, that's written. So there can be different combinations. So that's about dihybrid cross. The things that you have not studied, I am trying to teach you that as of now. Okay? Now, yes. So now you tell me the answer for this question. In a dihybrid cross in pea plants, Mendel got 9:3:3:1 phenotypic ratio. Phenotypic. Okay, not talking about genotypic ratio at all. Talking about phenotypic ratio, how they appear. It denotes that alleles of two genes are interacting with each other. It is polygenic inheritance. It is multiple allelic inheritance. The alleles of two genes are segregating independently. What is the answer? It will be D. The alleles of two genes would be segregating independently, my dear children. That was law of independent assortment. Now, who noted that behavior of chromosomes was parallel to the behavior of genes? Who was the person? So, Mendel just said a lot of things and he went. Okay, he said there is factors are basically going to govern each character and things like that, and okay, there were so many things. But some people came in and said that there were chromosomes and chromosome behavior was parallel to the behavior of genes, or the factors that they were talking about, he was talking about. So my dear children, when I say a chromosome is getting separated, don't you think I am also trying to say that a gene is getting separated? Because chromosome contains those genes. So who was the person who noted all this thing? Was it T.H. Morgan? Was it Gregor G. Mendel? Was it Alfred Sturgeon? Was it Walter Sutton and Theodore Boveri? Your answer for this question lies over here. Walter Sutton and Theodore Boveri. But you do not know it because you have not learned it. So I am going to teach you that. There are certain important things that you need to know. First is, when did Mendel do his experiment? 1856 to 1863. For 7 years he did his experiment. He came down with all the laws and things like that. Now after that it went away. But there was some time when Mendel's work was rediscovered. It was during the year 1900. Three scientists, remember the names, De Vries, Correns and von Schirmack. They independently rediscovered Mendel's work. Okay, 1900, in the year 1900. Now my dear children, in 1902, when Mendel's work was rediscovered, Walter Sutton and Theodore Boveri, there were two people who came into the picture, who said that, hey, the Mendel thing, he was talking about some factors being separated and things like that happening, in the gametes. Actually, the factors are nothing but the genes, and the genes are located on the chromosomes. Because whenever I am saying that genes are separated, I am also saying that chromosomes are getting separated. So, the behavior of chromosomes was parallel to the behavior of genes, and used chromosome movement for that was put down by Walter Sutton and Theodore Boveri in 1902, just 2 years after the rediscovery of Mendel's work. Now, they were the people who put down the chromosomal theory of inheritance, okay, who added chromosome into the story as well. Sutton and Boveri said the pairing and separation of a pair of chromosomes would lead to segregation of a pair of factors that they carried. That is, a pairing and separation of a pair of chromosomes would also lead to separation of what? The genes or the factors that they are carrying, my dear children. Later, chromosomal theory of inheritance was put forward by Sutton. In your NCERT is written that Sutton is the one who put forward the chromosomal theory of inheritance. Actually speaking, before it was written that Sutton and Boveri both were involved, okay, but later towards, if you read your NCERT, is written that chromosomal theory inheritance was put down by Sutton. So if at all in the question, if at all Sutton is mentioned separately, you can go with that. If Sutton and Boveri is mentioned and only that is mentioned, you can go with that as well. Just like how we saw over here. Who noted the behavior of chromosome was parallel to the behavior of genes? Walter Sutton and Theodore Boveri. Okay, but then when you say all this thing, you need some proof also. And who did that experiment to prove it? It was Thomas Hunt Morgan who did the experiment to prove what? The chromosomal theory of inheritance. Also remember, in this picture comes one guy whose name is Alfred Sturtevant. he was actually a student of Thomas Hunt Morgan. He was a student of Thomas Hunt Morgan, my dear children, and he also did his findings. So Thomas Hunt Morgan was a person who was experimentally trying to prove what Sultan and Bovary said. Okay, and helping him was Alfred Sturtevant. Now over here I wrote the answer. Just a second, okay. Now I'll look for a question linked with this. Okay, one question that is linked with this is, yeah. So I told Thomas Hunt Morgan tried to prove it. How did he try to prove it? By doing an experiment on fruit flies. Fruit flies, you call them as Drosophila melanogaster. What do you call this as? Drosophila melanogaster, my dear children. And why was this fruit fly being used for this particular thing? The reason being that they could be grown on simple synthetic medium in the laboratory. They were very easy to be grown. Okay, they complete their life cycle in about 2 weeks, just 2 weeks, whole complete life cycle would be done. A single mating could produce large number of progeny flies, so you can do multiple experiments, and there was a clear differentiation in the sexes. The male and the female flies could be easily distinguished. First thing, simple synthetic media could be used, they could be grown in that only. Second thing, the life cycle was small, so I don't have to wait for years to find out my findings. The third thing, they were producing large number of progenies, and the fourth thing, the sexes were clearly differentiable. That's the reason. And now, my dear children, comes to the conclusion of this particular thing, which is what are the conclusions drawn by T.H. Morgan from the crossing experiments in Drosophila in respect to linkage? So when he was finding that, he saw that okay, chromosomes, whatever he said was correct. Sutton and Bovary, whatever they told, it was correct. But apart from that, I also have a finding that is about linkage. There are certain genes which are very closely linked. When they are closely linked, you do not find new combination. When they are far away from each other, you find new combination. So higher the linkage, higher they are close, lesser is recombination, and lesser the linkage, higher is what? Recombination. In other words, genes which are tightly linked will show lesser recombination, and the genes which are loosely linked will show higher recombination. So with this recombination rate and stuff like that, his student Alfred Sturtevant, who was there, my dear children, he was able to analyze the distance or the positioning of what? The genes. That means if the genes are showing higher recombination, my dear children, they are what? Far away from each other. This is the distance between them. So this thing was later found out by Sturtevant. So that is also one finding. I am not able to find where have I written that. Wait. I have written it somewhere. No, it's not there. Okay, it's not there. But anyways, this is one very important thing that I want to tell you, that is about Alfred Sturtevant. He was able to find gene mapping. He was able to locate the gene on the basis of the percentage of recombination. If there is higher recombination, that means they are far away. The genes, I have been able to map the genes, they are far away from each other, like that. So that was done by Alfred Sturtevant. Cool? That was his finding. And apart from that, my dear children, something that is new, something that you need to know is this. Recognize the figure and find out the correct matching. What is this? And what is this? The male Drosophila is smaller in size. The female Drosophila will be larger in size. Please remember that. Also one very important question. I have given the answer, but still. How many type of gametes will be produced by individuals of capital A, capital B, capital small c and small c genotypes? Over here, you have to find how many type heterozygotes are there. Heterozygotes mean the one that looks different. So capital B, small b looks different, right? So you have a formula 2 raised to the power n. Number of how many heterozygotes are there, you have to put it over here. So 2 raised to the power 1, only one heterozygote is there. So 2 raised to the power 1, 2. So the type of gametes that you will be finding will also be 2. Okay, so these are the few questions that I do have. There are certain other questions, but I think with the time limitation, I should not be taking it forward. My dear children, I will be promising you that I will be starting off from this chapter when I go live for complete revision. Okay, I will start with principles of inheritance and variation so that you don't miss out anything. Anything which was new, which I didn't taught, I have already taught you here itself. The rest, everything is there in Sankalp 2 videos. Please go watch it. It is going to be helpful for you. And this is homework number 6 question. The law of segregation states that, you have to tell me what does it state. Law of segregation, this is second law, okay, not law of independent assortment. And with that we complete this. Okay, the reason that I need to complete, there is a specific reason I'll tell you later on. As I told, this is my promise, I will be beginning from principles of inheritance and variation in the live session, and you have to be there for the live. Okay, we will complete it together. And if at all you have not subscribed to the channel, definitely do not forget to subscribe it right now. Wishing you all the best. Please take care of yourselves. I am here to help you out, and please put down your doubts in the comment section below. Bye bye, take care.